Hello, this is Brian. I'm your instructor for the How to Write Your Scientific Journal Article in 10 Weeks course. This video is an introduction to the course, and I'm just going to tell you a little bit about what the course is about and what to expect, and give you some context for how I designed this course. I'd like to start off with this comic from phdcomics.com. It's kind of funny, but it's also illustrative of the problems that a lot of scientific writers have. So this comic shows what they call the research cycle. So in the first panel, we see this PhD student, and he's at the beginning of the research cycle, and he's reading. Not only is he reading, but he is very frantically reading. You can see behind him, he's got stacks of journal articles. He's got one article in each hand. He's trying to read them both at the same time. He's also got a book open in front of him. And you can see he looks pretty stressed and anxious. The step 2 of the cycle is write. And you see he's frantically and ferociously typing away at his keyboard. Part 3 of the cycle is rinse. He's drinking a coffee shop coffee in one hand, and he's got a homemade coffee and a full pot there in front of him. And then the cycle starts over, repeat, and he's back to reading. If you fall into this kind of a cycle where you're just, frantically trying to do all these things, that's not real productivity. And it's not very relaxing. It's very stressful. And so what I want to do in this course is give you a system where you don't have to fall into this kind of a cycle that's very stress and anxiety producing. So over my years of teaching scientific writers and talking to scientific writers as a mentor and a consultant, I have come to know pretty well what the common problems are for scientific writers. The first one is you have not been trained in scientific writing. Maybe you've had a workshop. You probably haven't had a full course like you're going to get in this course that teaches you about scientific writing in a systematic and step-by-step method. And number 2, your training has been wrong. So you have probably taken some writing courses during your undergraduate experience, composition and rhetoric, or maybe a creative writing course. The kinds of techniques you learned in those courses are correct for composition and rhetoric or for creative writing, but they're not correct for scientific writing. In fact, they're counterproductive for scientific writing. And so that training may actually be hurting you as a scientific writer. Number 3, you have been expected to learn to write just by reading scientific papers. And this might work for a small percentage of people, but for most of us it's not going to work. So think about it this way. You've probably listened to a lot of songs. Does that mean you can sit down and write a song? Same thing with movies. You've probably watched a lot of movies in your lifetime. Does that mean you can sit down and write a screenplay? Probably not. There's a lot of subtleties that go into writing these works that you can't pick up just from being a consumer. And to me, it's the same thing for writing. I myself had read hundreds of papers. I'd even written a lot of papers before I really learned how to do it properly. Problem number 4 that a lot of writers fall into is that they're trying too hard to sound smart. So they read these scientific papers. The papers are using specific terminology, and they're using these big fancy words. And the writers feel like, oh, if that's what I'm seeing, then that's probably what I'm supposed to do. And so they make an effort to recreate that kind of voice or sound. And if they're not able to do that to their expectations, they feel like they're not doing a good job. Well, I'm going to tell you to let that pressure off yourself. You don't have to use big fancy words. and sound professional or sound academic to write a good scientific paper. There are research studies that show that plain language is better than jargon and fancy terminology. Okay, point number 5. The reason it's so hard to write is you don't know the secret formula. And the reason I have secret in quotation marks is because it's really not a secret. It's just something that is not obvious unless you have been told about it. So what formula am I talking about? The way to structure your arguments or your progression of logic such that the scientific paper flows. There is a way to structure this progression that makes the writing process much more easy. That's what I'm referring to by the secret formula. Point number 6, your skills are weak. Most of people that would be taking this course are early career researchers who don't have a lot of practice. If you don't have a lot of practice, then naturally your skills are not going to be that developed. You have to get feedback and practice and awareness and develop your skills over time. And this course will help you with some of that. I also have a companion course that's about scientific writing skills that goes into more detail on a lot of the skills that are used in scientific writing. Number 7, one reason that it's so hard is that you feel intimidated. And this is completely understandable, and we all go through this. When you're first starting out and you're going to publish a scientific paper and release it into the world for everyone to see, it is a little scary. But I'm here to tell you that everyone feels that way. We all had to start at the beginning, and your ideas are good. You've discovered some new things that no one knows. And for those new things to be useful to the scientific community, they need to be released into the world. They need to be published so people can know about it and use that knowledge. This course will help you feel like you produced a good product, and it will allow you to feel less intimidated, and you can push on through that and go ahead and publish your paper. Reason number 8 is you have no idea where to start. There's so much to write. Where do you start? In the course, I'm going to give you a system for working your way through the paper, and it's worked very well for students that have taken my course. And point number 9, one of the reasons it's so hard is that you misunderstand the purpose of writing a journal article. A lot of times new authors will feel like the purpose of them writing a journal article is for them to prove themselves. And that's really not the point. So all of your readers are going to assume that you know what you're talking about. The purpose of writing the paper is to release your new knowledge into the world, into your field, so that the other people, your colleagues in your field, can use that new knowledge for their research and to expand our understanding of the world. I have another comic from phdcomics.com, and this one shows the neurobiology of writing. On the left, how it's supposed to work. They just think with their brain, which executes a command, and then their brain processes the language, controls their hands, transmits the command to the muscles, which type onto the keyboard, and the words appear in the computer. Kind of like a computer itself or a robot. But how the person usually feels is they have panic, fear, insecurity, confusion, leading to no motivation and hesitation. What I'm trying to do with this course is take all of those negative emotions and decrease them substantially. This is a quote I found from Tim Albert. The quote says, "It works like this. The author, usually the youngest member of the team, is given a huge amount of data, but no training, no resources, and no extra time, and is then expected to produce a first draft. When he or she finally finishes the first draft, the co-authors are encouraged to make as many criticisms as they can, or even to redefine the task completely." true. This is a true experience for a lot of beginning writers. This is part of the purpose of my course, is to give you the training and resources to take that huge amount of data and produce a first draft. Here's a quote from William R. Llewellyn. "I do not believe it is possible for one person to teach another person how to write." You might be thinking, why am I telling you this? This is a writing course. Why do you tell me that you can't teach me how to write? Well, this is not the complete quote. Let's look at the rest of it. "One person can teach another about writing, but the only way to learn how to write is to write." Okay, now it makes more sense. Let's dig a little bit deeper. What you see on this slide is what I call the writing improvement cycle: awareness, practice, and feedback. During this course, I'm going to help you develop a lot of awareness about scientific writing, and between lessons, you're going to be engaging in practice. And then you'll need to seek feedback. So some of the feedback can be self-feedback, but I also encourage you to find a friend or a colleague who's willing to read your work and give you feedback. And as you get the feedback, that's going to increase your awareness. And as you continue to practice and get more feedback, then the cycle will continue and your writing skills and your writing ability will continue to improve. The writing improvement cycle applies to reading and to editing and revising. Now let's talk about my goal as your instructor. My goal is to help you write your scientific journal article in the most efficient way possible. My objectives are to help you become aware of good scientific writing structure, to help you develop a project management system for writing, to help you set up a plan to write your paper efficiently, to give you step-by-step guidance for writing your paper, and to help you become a better scientific writer and scientific thinker. Okay, one last PhD comic. So the student says, "It's a new year, time for a bold resolution. I hereby declare this will be the year I write my thesis." Then she types, "my thesis." And she's thinking, that was easy, now what? No, this student is not using a project management approach to writing her thesis. How do I know that? Well, she has no plan. We can tell that because she says, that was easy, now what? So she doesn't have a plan. And clearly she doesn't have milestones. So if she had a plan with milestones, she would know exactly what to do next. We can also tell from this comic that her project is open-ended. It doesn't have any deadlines because she's saying, oh, I'm going to do it this year, which implies that she could have done it last year, or she could wait and do it next year. You know, having her research and thesis writing be so open-ended and drifting aimlessly is a recipe for stress and misery. And I want to help you avoid that stress and misery by giving you a project management approach to write your scientific paper. So a project has 5 process groups or key elements. There are milestones and deadlines that are built into the process. And so I have a graphical representation of a project. We have a start point and a finish point. So there's first the initiating stage, which leads to the planning stage, leading to the executing stage. And while executing is happening, we're also looking back at the plans, and we're controlling and monitoring the progress of the project. And so by controlling, it means we make adjustments to adapt, to keep on track with the milestones. And by monitoring, it means we're looking at the plans and making sure that we're on track. And so once you've finished executing, it's time for the closing stage. And this is where we're going to get the paper ready to submit to the journal, and then we will finish. So that's going to be a great feeling when you get to that finish line. Let's look at the course structure. So the course is built into 10 stages or modules The first stage is how to read scientific papers. It's probably going to take you about half a week or maybe a full week to get through this module. The second one is literature search. That should take about a week and a half. Then we'll go into pre-writing for about a week, and then we'll spend about a week on the introduction. We'll go to materials and methods. That should be half to 1 week. The results probably take you about a week and a half. Discussion should probably take about a week and a half. Then we'll do the title and the abstract. We'll look at editing and revising. And to close out the project, I'll talk about how to submit your manuscript to a target journal. So let's break that down a little bit more and look at those different stages of the course on a project management flowchart. So we see in the initiating phase, we have the scientific paper structure. In the planning stage, we build the reference library and we do some pre-writing. The executing phase is going to be the bulk of the course, where we write the introduction, materials and methods, results, discussion, and title and abstract. And then in the closing phase, we will edit and revise the papers and submit to the target journal. Each lesson is going to conclude with an action list, and the action list is going to be your step-by-step set of instructions that will tell you exactly what to do to write your paper. So make sure you follow that action list each week. Finally, let me give you some tips on how to do well in this course. The first thing I would suggest is to always do the lessons. And I think it's really important that you finish one lesson before you move on to the next one. But to really spend some time thinking about what we're talking about, thinking about the papers that you're reading, and reflecting on the process of what you're doing. Thirdly, and this seems obvious, but put a lot of effort into the writing and the revising. Your first draft is not going to be your final draft. So keep that in mind. It's normal to go through quite a few drafts, and that's going to be part of the process. The next point I would make is to write every day, at least a little bit. Now, I'm not saying you need to spend 4 or 5 hours every day, but I do find that consistency is very important. The next tip I have is to write in short sessions, not in binges. So that goes along with writing every day. So it's better if you write for 1 hour, Monday, Tuesday, Wednesday, Thursday, and Friday, than just writing for 5 hours on one of those days. The next one is don't procrastinate, just get it done. So just sit down and write, no matter if you don't feel like it. Sit down and get to work and write. The next tip is to read and cite a lot of papers, but you're going to need to read a lot of papers to write a good paper that's thorough. And lastly, I would say have fun. But if you don't, at least enjoy the challenge and savor the satisfaction of completing your manuscript. All right, well, that's what I have for the introduction to the course. Now you are ready to start on lesson 1. Happy writing! Hello, and welcome to lesson 1. This lesson is about scientific paper structure. Now, let's talk about where this lesson fits into the overall scheme of the project management approach to writing your scientific paper. So we are in the initiating phase, looking at scientific paper structure. And after this lesson, we'll start moving into the planning phase before we get to the rest of the project. The learning outcomes for this lesson are as follows. At the conclusion of the lesson, you should be able to first explain the purpose of each section of a scientific journal article. You should be able to explain the overall structure of a scientific journal article. You should be able to identify key elements or notice and identify when there are missing elements or obscured elements in a published journal article. And finally, you should be able to outline your journal article. Let's talk about how scientific papers are structured. Most papers in most journals follow the IMRAD format. So what does IMRAD stand for? IMRAD is an acronym. It stands for Introduction, Materials and Methods, Results, and Discussion. So let's talk about the purpose of each of these sections of the journal article. The introduction is there to provide the context for your work. It's going to let your readers know where your work is coming from. It's going to let your readers know how your work is connected to previous research. It's also going to let the readers know what the purpose or the hypothesis of your study is going to be. And so it sets up the expectations that they have for the rest of the scientific article. It's an extremely important section to get right if you want to write a coherent and cohesive journal article. The methods or materials and methods section is there to let your readers know how you did your experiments, what materials you were using, and if it's not obvious, in this section you will explain why you chose certain methods to carry out your experiments or your research project. The results section is there to interpret the data. So data is not equal to results. Results are data that have been interpreted in the context of that experiment or that part of the project. So the results section is also there to help the readers see how you've interpreted that data, and it's going to present the figures and tables in a way that they follow it as the narrative of your paper unwinds. Then the discussion section is there to interpret your results. So you're interpreting the results in the context of your field and other previous research that has been done, and also in the context of your research question, that is, your hypothesis or your purpose statement for this particular study. Also in the discussion, you're going to discuss the implications and significance of your findings, and you're going to include a critical analysis of your study, its strengths, its weaknesses, the unique aspects about it. You're also going to look into the future and say what should be done next. So at this point, I would like to step back a little bit and just think about what we're doing in science. So in science, we follow the scientific method. And you can see in this figure, which I've pulled from this scienceschools.us website, a very simplified version of the scientific method. Now this figure was meant to educate children, and you probably saw a very similar figure when you were a child, first learning about science. And so this is how the method goes. First, you ask a question. Then you do some background research, construct a hypothesis. You test the hypothesis by carrying out an experiment. Then you analyze the results and draw a conclusion. Now, this conclusion will either support your hypothesis or it will not support your hypothesis. So you can say the hypothesis is true or correct, or it is not correct. If the hypothesis is not correct, then we have this loop where you go back and you rethink the hypothesis, you change it with your new knowledge that you have, and then you test it again, you analyze the results again. Once you have an answer, it's time to report the results. Now, this is a diagram for a hypothesis-driven research. Not all scientific research is hypothesis-driven. Some of it is driven by technology development and other kinds of development types of studies, knowledge discovery, for example. And a hypothesis just doesn't work for those types of inquiries. So we'll have another structure as well. And this structure is for those purpose-driven types of experiments. And it's very similar. You notice how things are now, and you identify a need. You do background research, you propose a new method or technology, you develop a way to solve the problem, and you test that method or technology. Then you analyze the results and draw a conclusion. Either it works or it didn't work. If it didn't work, you go back through this loop to propose a new method or technology and go through the process again. So it's very similar to the scientific method, but it doesn't use a hypothesis. So having been a scientist for a long time, I can say that this chart is pretty much accurate for the way we work. Now, we don't think about it necessarily in these terms as we're going about our jobs as scientists. But I think when it's time to write the paper, it's good to think about this simple scientific method again, because it corresponds to the way that journal articles are structured. They match each other very well. Well, the introduction covers the first few parts, the asking a question, the background research, and the constructing the hypothesis or purpose statement. Materials and methods and the results covers this green circle where you test with an experiment, and then the discussion covers the rest of it, analyzing the results to draw conclusions, discussing whether the hypothesis is supported or not, and reporting the results. One of the things I want to do in this course is help you write a good scientific story. And when I say story, I don't mean fiction. I'm not at all encouraging you to make anything up or cherry pick your data and leave out things that are inconvenient. No, I'm not saying that at all. By story, I mean a true story, a true and complete story that is interesting. It has a narrative. It moves from one step to the next step to the next step, and people are interested in learning about it. That's what I mean by a good story. Let's talk about how to structure a good scientific story. It's going to set the context in terms of current knowledge. It's going to clearly define the unknowns or the knowledge gaps that will be addressed in that study. And it's going to let the reader know what to expect by having a clear research question. It's going to be simple, but not simplistic. As scientists, we really like to overcomplicate things. It seems to be in our DNA. But when you sit down to write your paper, make sure that you're not overcomplicating things. Simplify it as much as possible so that you can have a nice, simple story. Additionally, a good scientific paper is going to be concise. That means it's no longer than it needs to be. It doesn't necessarily mean that it's short. It just means that there's no excess words, no excess verbiage in the story. It's as short as it needs to be and as long as it needs to be. Also, a good scientific story is going to answer the research question that was posed earlier, and it's going to fill in the knowledge gaps or the unknowns that were mentioned earlier. And lastly, it's going to identify new knowledge gaps. So this drawing shows a funnel on top of a flask. And so basically this is making up a pyramid shape. This pyramid shape is going to be a graphical representation of the structure of a scientific paper. So I'm going to redraw this hourglass shape and talk about how to tell a good story in your scientific journal article. The top part of the pyramid represents the introduction. And you see it starts out very wide at the top, and as you go through the introduction section, it becomes more narrow. What this represents is the way you're presenting information. At the beginning, it's going to be more general, and as you move through the introduction, you're going to become more and more specific in the information that you are presenting. Now, the first few paragraphs of the introduction are going to establish the current knowledge in the field. And you're going to do that by using knowledge bricks or little bits of information about what's known and supporting that information with references. And so you're going to stack these knowledge bricks together to build up a wall of knowledge. So references 1, 2, 3, and 4 are used to build part of the wall of knowledge. References 5 through 9 are used to build another part of the wall. And then 10, 11, 12 are building another part of the wall of knowledge. Now what you notice is that between these parts of the wall of knowledge, there are gaps. So there's information that we don't know as a field. These are the knowledge gaps, or the unknowns. And so the purpose of the introduction, one of the purposes, is to point out these unknowns or knowledge gaps. You'll have one or maybe two in your introduction. And then you're going to design a research question that's going to fill those knowledge gaps when you answer that research question. And so the knowledge gaps should logically lead to the research question. And to carry out the work of answering the research question, you'll break it down into some objectives. The next part of the scientific journal article would be the materials and methods section, followed by the results section. And those are represented by the straight part of the funnel, because they're relatively straightforward. You're not going more specific or less specific during these sections. So the materials and methods should be presented in a way such that they match up well with the objectives that were proposed at the end of the introduction. And then they should flow into the results. Now these results should reflect back to the objectives that you pointed out in the introduction. That's what this red arrow is representing. And after the results section, we will move into the discussion section. The discussion section should start out very specific, and that's why it's narrow at the top. And then as you move through the section, it's going to become more generalized. That's why it's wider at the bottom. The first part of the discussion section should present the major new knowledge that you have found, and that should reflect back to the objectives and the research question that were stated in the introduction. As you move through the rest of the discussion section, you will discuss other new knowledge that you have found, and you will talk about how that new knowledge relates to the knowns that you talked about in the introduction, how it fills the knowledge gaps, and how it creates new understanding in your field. Another way to present this information is on this form that you're seeing now. This is the scientific paper reader-writer outline form that I developed in 2014. Now I designed this form for people to use as they're reading scientific papers to capture the information that they're reading on a one-page form. But you can also use it as you're writing scientific papers. This is kind of a way to put your thoughts together in a logical sequence. So the form starts in the upper left corner with the background, and then just follow the arrows. Once you write a few bullet points about the key background points that you need to present, those are going to lead to the knowledge gap or the problem to be solved. That's going to lead to the research question or the purpose statement for the study. And that's in a bold box because that's really the heart of the paper. What you can do is start with the research question or purpose, then work back to the knowledge gap, and then work back to the background and say, what background do I need to present to show this knowledge gap? After the research question or purpose, you're going to talk about the hypothesis or the objectives. You'll talk about the approaches that you use, the results, the key results that were found, the conclusions, and then the conclusions splits in two different directions into unanswered questions or new knowledge gaps that we have discovered in the process of doing this project. And it also goes back to answer the research question or purpose and address the problem or knowledge gap. So you have this form as a downloadable as part of your course materials. Moving out of graphical representations, another way you can look at the structure of a scientific journal article is by using a classic outline. And so this outline, we structure it with Roman numerals and then break that down by A's and B's. So we see that in the introduction section we have A, B, C, and D. So A is the first background paragraph, B is the second background paragraph, C is the knowledge gap paragraph, and then D is the objectives and significance paragraph. Now your introduction might have more than 4 paragraphs. If so, then just add a third background paragraph to this outline. But basically in each background paragraph you're going to have your knowledge bricks and your references, and then in the knowledge gap paragraph you'll have some signals or signposts to the readers that show them that you're introducing a knowledge gap. And then also the research question will come in that paragraph. You should also propose your hypothesis or purpose, list your objectives, and optionally give a findings preview and a significance. This outline continues. Part 2 is materials and methods. So we break that down into materials, method 1, method 2, and for each method you need to make sure you include whatever documentation of required approvals that you might need for certain protocols or project methods. Part 3 would then be results, and it's a good idea to break the results down by a 4-part repeating pattern where you discuss the background, the purpose, the results, and if necessary, an interpretation of the result. And we'll get into more detail in the results lesson about what I mean by interpretation of the result. Part 4 is the discussion, and that's going to be broken down into the answer to the research question, the major findings and the relationship to previous research, critical evaluation of the study, generalization and significance, and conclusion and future directions. All right, so we have reached the end of the lesson. I'm going to give you your action list for this week. The first thing you're going to do is to read 3 papers from your field while filling out the reader-writer form. As you read the papers, write down the bullet points for the background and all of the other key elements. Now, if those key elements are hard to find, then the authors didn't do a good job at showing those key elements. So they might be missing, or they might be written in a way that they're obscured. They're not clearly shown. So notice that. You're not necessarily going to find every element in every paper that you read. If it's not there, then think about how they could have written it differently to do a better job at showing all of these key elements. Action item number 2 is to take 10 papers from your field, pull them out, and just start counting the number of paragraphs in each section. Make a spreadsheet, an Excel or Google Sheets, and keep track. How many paragraphs are in the introduction? How many paragraphs are in materials and methods? How many paragraphs are in results? How many in discussion? And then I want you to calculate the average number and look at the range. What's the fewest number of paragraphs? What's the most number of paragraphs? And what this is going to do is give you kind of a benchmark. It's going to help you know what is normal in your field. So that can give you kind of a target for how long each of your sections should be. Action item number 3 is I want you to count the number of references for 10 papers in your field. Again, I want you to calculate the average and look at the range. So what's the normal number of references that are cited for papers in your field? The 4th action item is to read 3 papers that are outside of your field. So go and find 3 papers that are a completely different topic than what you look at. If you research animals, go and find some papers about plants. If you research plants, go and find some papers about geology, or whatever you decide to look at, but just something that's completely different than from what you do. Because what I want you to do with these papers is to read through them and find these same key elements that you were looking for in the papers in your field in action item 1. Doing it outside of your field is a useful exercise because now you're looking at the way that the authors structured their papers, and you're not getting distracted by the scientific details. You're not thinking about the science aspects. You're just thinking about how the paper was put together. And the final action item is I want you to reverse outline 2 papers. So by reverse outline, I mean just take that outline, the classical outline that we looked at a few slides ago, and as you read these 2 papers, go through each paragraph and make an outline of how the authors structured their paper. So this is going to help you think about how papers are put together. It's a companion exercise to the reader-writer form, but it's a little bit different. You're looking at the bones of the paper, the structure of it, how the authors built that paper. So I really encourage you to do all of these action items before you move on to the next lesson. Let's wrap up by looking back at our learning outcomes. I want you to make sure that you can do each of these learning outcomes before you move on to the next lesson. So sit down with a friend, ideally a friend who's not in the sciences, and explain to them the purpose of each section of a journal article. Explain to them the overall structure of a scientific journal article. Make sure you can identify the key elements or notice when they're missing in published journal articles. And then you should be able to use this information to take the reader-writer form or the classical outline and begin to outline your paper, the one that you are writing during this course. Okay, that's all I have for today. After you have completed the action items and you can do all of the learning objectives, then you are ready to move on to lesson number 2. Most of all, happy writing! Welcome to the lesson on finding and citing references. The learning outcomes for this lesson are as follows. At the conclusion of this lesson, I would like for you to be able to do the following things. First, explain the purpose of citing references. Secondly, be able to identify appropriate sources of references and be able to avoid citing inappropriate references. Third, I would like you to be able to correctly cite references in your text and in your bibliography or references cited list. And the last one is you should be able to find references in databases and import them into reference managing software. All right, so let's get started. Why do you need to cite references? Anyway, what's the point of citing references in your scientific paper? Well, there are a few different reasons that this is important. The first one is that you need to give credit to those who originally made the discoveries that you are referencing. So just like when you publish your papers, you want authors in the future to cite you when they're talking about your work and how it's important, you need to do the same thing for other authors. So it's just part of the system that we have in science for crediting authors for their contributions. Another reason to cite references is to help your readers know where to look if they want additional information or if they want to verify what it is that you are writing. In other words, references are a way to direct the readers to specific literature. And another reason to cite references is to reassure readers that you've done your background homework, you know what you're talking about, and that your premise is valid, it's scientifically rigorous, and it has a basis in previously published work. So those are the basic reasons why you need to cite references in your scientific journal article. This is a quote I like to show from Samuel Johnson. He says, "The greatest part of a writer's time is spent in reading. In order to write, a man will turn over half a library to make one book." Now, this doesn't apply only to men, obviously. It's the same for women. So a human will turn over half a library to make one book. The point of me showing you this quote is to show that you're going to have to do a lot of reading to write your paper. I mean, you're not going to sit down and write it in isolation. You need to know what has been done before so that you know how your work fits into that context. And so you'll read a lot of papers as you write each of your scientific papers. Now, in terms of references, what types of references should you expect to be citing? So we can refer to references or source material as primary sources, secondary sources, tertiary sources, and there are other sources that can get you started, but they're not acceptable to cite in a scientific paper. So let's look first at primary sources. Primary sources are the original peer-reviewed publications, usually journal articles, but sometimes they'll be published in book chapters. These primary sources will be the bulk of your citations. Secondary sources are review articles in scientific journals. If you're not familiar with a review article, that is an original publication in a scientific journal summarizing and analyzing other literature that has already been published. For example, you might have a review article about diabetes. The authors would read hundreds of papers and they would describe the results from those papers and put it into a logical framework and draw some conclusions about that body of work. And a good review article will come up with new questions, but sometimes review articles are just a summary of what has been done up to that point. Sometimes it's okay to cite these review articles, and we'll talk about when you can cite these secondary sources. Tertiary sources will be cited occasionally. It's not very common. Most journal articles will not have any tertiary sources. But sometimes you will use a tertiary source. So what are these tertiary sources? They include newspaper articles, textbooks, and official or scholarly websites, such as databases from the FDA or rules and regulations from FDA, statistics from the CDC or USDA, World Health Organization, those kinds of facts and figures that you cannot find in a primary journal article. Now, sources like Wikipedia and encyclopedias are not considered to be valid scientific sources. Now, I'm not saying that you should never go look at Wikipedia, because Wikipedia is kind of like a review article in itself, and it can get you started because the articles on Wikipedia typically will cite specific primary sources. So it can be a good way to get started, but you shouldn't cite Wikipedia itself as a source in your journal article. So where do you find these sources, the articles that are going to become your references? One place you can look is the key papers that are related to your work. So you probably have a stack of papers that you are reading as you're doing your research. You can take a look in those papers and see the sources that they are citing. There's a good chance that if it's a source for work in a paper that's closely related to the work that you are doing, that it would be an appropriate citation for you as well. Check there. That's one first place you can go quickly to find some references. Secondly, you can just keep your eyes open as you are browsing. Look for announcements, press releases, news articles. You can look on the professional societies' websites. They'll talk about research. If you look at social media like Twitter or LinkedIn, you'll see articles being promoted by their authors or by people that just think it's interesting work. Sometimes I've found a lot of good sources that way. You can look at science news sites, and they will be summarizing research in their articles. You can also search databases. And this is going to be the major way that you find most of your sources. The common databases are Google Scholar, Web of Science, Scopus, and PubMed. And there may be a few others, but these are the big ones that most people are going to be searching as they're looking for references or source material. So now that we've talked about sources of references, let's now discuss what references are appropriate and how you know which reference you should cite in a particular situation. So let's look at an example where I need to cite a primary source. I am going to cite a specific original finding that is critical to my argument. In this example, I want to establish some background knowledge, and that background knowledge is that IRT1 was the first iron transporter gene identified from plants. So that's a very specific statement. I should cite the original paper that first discovered this knowledge, and you can see the citation here, ID et al. 1996. Now this 1996 paper has been cited in a lot of places. It's been cited thousands of times. So could I cite a review article that makes this statement, such as Waters and Sankaran 2011? No, that is not appropriate. That's a secondary source, and I'm making a very specific statement, so I need to cite a primary source. So I should not cite a review article. Now, what about citing a newer article that mentions this finding in its introduction? Can I cite that? For example, Stein and Waters 2012. Absolutely not. Don't do this. I bring this up because as a reviewer, I started to see this a lot. Authors that were citing other primary papers, using them as a secondary source for a very specific statement. You shouldn't do that. You need to trace back through the literature to the very first original article that discovered this knowledge that you're trying to cite. So here's another example. I want to cite a general finding that's critical to my argument. In this case, I want to say that iron transporter genes are critical for plant iron uptake in multiple species. So that's a general statement. It's not talking about one specific iron transporter. It's talking about a whole category. So I can cite secondary sources for this. If only a few papers have established this fact, what I'll do in that case is cite them all. So if it's less than 4, I'll just cite all 4 of those. If it gets to be 5 or 6 or 7, in that case, I'm going to go over to a review article and cite a review article. Now, in the case that there have been no review articles that are appropriate to make this statement, and there's more than 4 or 5 papers that have been published, then I will just choose the first 2 or 3 chronologically and cite those papers. Now, when you are citing sources, make sure you do your due diligence. What do I mean by that? You need to verify the information in those references, especially if you're getting the references from a previous paper. And you say, well, I'm making the same point that this previous paper is making, so I will cite the same references that this previous paper is citing. That's fine to do that, but you need to verify that those previous authors got it correct. So you need to go into those papers that you're going to cite and make sure that they say what you think they say. Okay, so you need to read them, or at least read them enough to verify the information. Because the accuracy of the references is your responsibility as the author. You also need to make sure that your paper's reference list at the end of the paper matches up with the in-text citations. And so when I first started writing papers, I didn't use the reference managing software, and I had to do that manually. So I had to print my reference list and print my paper and trace through the paper, and every time there was a citation I needed to go to the reference list and make sure that they were accurate with each other, that all of the references were accounted for, none were missing. Now I really recommend that you use reference managing software. That makes this easy. Then your paper's reference list is automatically going to match up with the in-text citations. Also, make sure that the references are showing up in the list in the correct format. So different journals are going to have different specifications, different styles for the way they want you to write formal citation. So they might ask you to have a certain capitalization. They might ask you to use italics for the journal names. They might want you to abbreviate the journal names or not abbreviate the journal names. So again, if you are using reference managing software, this is very easy because you tell the software which journal you're going to submit to, and it automatically formats all the references. However, be aware that when you import references from databases like Google Scholar or Web of Science, there might be errors in the format that you need to correct. And you need to correct those in your database, not in your Word file. If you correct them in your Word file, the next time it syncs with your reference managing software, it's going to revert back to the information that's in the reference managing software's database, and will overwrite all the changes you made in Word. Where are you going to be putting references in your paper? This table shows you where they're going to go. In the abstract, there are no references, typically. The introduction will have a lot of references, so will materials and methods. Results may have no references, or it might have a few. It won't be very many if there are references in the results section. And the discussion section will have a lot of references also. Where do you drop the in-text citation within your sentences? What I recommend you do is to go and look at the papers that you're reading and see how they do it. There are natural places where you would want to drop the references. Sometimes it's in the middle of the sentence. It's not always at the end of the sentence, so be aware of that. You might have a sentence that is a compound sentence with different phrases that need citations for each of those phrases. And so you might drop the references before you get to the end of the sentence. So get a feel for how it's done in your field. Now, I already mentioned reference format and that it differs by journal. So what you need to do is, before you start writing, figure out what your target journal is and then go and look at their instructions. So the references could be author year, they could be a numbered list, they could be a numbered list that is shown as a superscript. It really depends on the journal and how they ask you to do it. Your reference managing software will put all the references in the correct format and in the correct order, so make good use of that. So by this time you should have an idea that you're going to be citing a lot of references in your scientific journal article. How do you manage these references? How do you remember which reference to cite in which place? There's a lot to keep track of. So you need to have a system for managing your sources. Your reference managing software needs to be an important part of that system. So you're going to use this software to build up your reference database. Some examples of the software I'm talking about are EndNote, Reference Manager, RefWorks, Mendeley, and Zotero. Which one do I recommend? Well, I don't recommend any of them over any of the other ones. They're all good. They all do what they're supposed to do. So just choose one that you like. If you are student, faculty, or staff at a university, check with your libraries because there might be some free reference managing software that you have access to. Now, what I recommend that you do is to build your database as you find your sources. So as you're searching Google Scholar or Web of Science or PubMed, whenever you find a paper that you might want to cite, go ahead and put it into your database. It only takes a second or two. It's going to save you a lot of time to go ahead and put it into your database now, rather than having to come back later where it's going to take you maybe 5 or 10 minutes to search again and find that paper again. And you need to figure out a way to keep track of which paper was the source of your information by developing a record-keeping system. Now, some of these reference managing software packages have a place where you can write notes. Some of them have a place where you can attach a PDF of the journal article along with the reference. So you can make use of that to keep everything in one place. If you don't keep track of your sources in your reference managing software, make sure you find another way to do that. So it might be something as simple as an Excel file where you take notes. Maybe you write notes on the paper itself, or some other way. There's different people have different systems, but make sure you develop one that works for you. Now, when you start writing your paper and you write a sentence that needs to be cited, I suggest that you go ahead and plug in the citation right at that point during the writing process. That's going to save you time later. An alternative to that is you can leave a comment. You could leave your comment in parentheses, or if you're using Word, you could make an actual comment box and put the comment there. What you don't want to do as you're leaving comments to plug in references later is to write something that's snarky or editorial about the paper. Just say which paper you're going to cite. Now let me show you an example of why you shouldn't leave these kinds of comments. This is a tweet from Hugh Kearns, and he has this example of a paper that was actually published where the authors left a snarky comment saying, "Should we cite the crappy Gabor paper here?" And they forgot to take that comment out. And it went through review that way, and it went through preprints that way, and it got published that way. And so that's pretty embarrassing for everyone involved. Also, you want to make sure that you're going to avoid committing plagiarism. And managing your references is a big part of that. What you might be tempted to do as your record-keeping system is to copy and paste from your sources into your document with the intention of coming back later and rewriting those sentences. But what if you forget to rewrite those sentences? Then you end up plagiarizing those articles unintentionally. So what is the definition of plagiarism? It is copying word for word, but it's also copying with some wording changes, like the adjectives or maybe the order of certain words, or you put in synonyms for certain words. That can still be considered plagiarism, even if you're acknowledging the source. Quoting material without acknowledging the source is basically copying word for word. Also borrowing someone else's ideas, concepts, and conclusions and passing them off as if you thought of those things. Even if you reword it, that's considered plagiarism. And summarizing and paraphrasing another's work without acknowledging the source is plagiarism. So as you're gathering references, it's really important that you paraphrase in your own words what it is that is important about that reference for you to cite. And that will help you avoid plagiarism. Now, there are some exceptions. If something is common knowledge, you don't necessarily need to cite a source. For example, if I'm going to say that the United States is in North America, I don't have to cite a primary or secondary source for that because that's common knowledge. DNA is the genetic material, is common knowledge. I don't need to cite a source for that. Also, in the materials and methods section, you might have some sentences that are word for word the same as sentences in other papers. I'm not telling you to copy and paste those, but some methods are just, there's just not very many ways to say certain things, and so it might end up with the same wording. That's not necessarily considered plagiarism. All right, so we are down to the action list for this week. The first thing I want you to do is to watch the other videos for lessons on how to search databases and cite references. So I'm going to have some demo videos showing you exactly how to go through the databases, how to get the references from the database into your reference managing software, and how to get it from your reference managing software into your Word document. Secondly, I want you to perform your database searches. So even if you have already built up your library partially, this is a good time to go back and do another search and see if you can find some additional articles either that you missed before or that have been published since the last time you searched. And build up your reference library. I want you to read papers and glean information. In this case, I want you to determine which papers you're going to cite. So you won't cite every paper you read. You might read a paper and then decide, well, this one is just not appropriate for this particular journal article. I might cite it in my next paper though. So determine which papers you're going to be citing. Then I want you to think about how those papers fit together to build your knowledge wall. These are going to be your knowns that you put into the introduction to build up your wall of knowledge. And I want you to recognize where the gaps are. These are going to be your knowledge gaps or your unknowns that you will point out in the introduction section. So let's review the learning outcomes that we looked at the beginning of the lesson. Again, please don't move on to the next lesson until you have completed all of these learning outcomes. So be able to explain the purpose of citing references, identify appropriate sources of references, and avoid citing inappropriate references. Know how to correctly cite references in your text and in your bibliography. And then find references in databases and import them into your reference managing software. All right, so this concludes this lesson, and happy writing. Welcome back, students. In today's lesson, this is going to be a short lesson. I'm going to show you how to find sources in Web of Science. I showed you Google Scholar before, and now we're going to look at Web of Science. It's a little bit different, much the same, but you can usually find different articles with the same search. So usually I use both Google Scholar and Web of Science when I'm searching. So the first thing I'm going to do is pull down this menu and switch it to all databases. So this will give us the most comprehensive set of results. It pulls in more journals than the core collection. And so what we're going to do is we'll start here with the basic search, and we can search by topic. And let's do a similar search to what we did last time. So we're going to search earthworm as topic, and just click search. And we got 44,000 results. So on Google Scholar, we got 47,000. We got 44,000 here, so still too many to start looking at title by title. So we need to narrow the search to make it more specific. And so again, what we were looking for is the effect of earthworms on plant root growth. And so when I use Web of Science, I like to start with just a very broad search term, because then when I add terms, it's refining those first results. So now I'm going to add plant and search that. We went from 44,000 down to 13,000. Okay, so that was a big cut. And now I'm going to type root. That took it down to 2,000. Okay, it's getting more manageable. And add the word growth. Okay, so now we're down to 1,000 results. Not too bad. That's including all years back to 1900 or something like that. So if you remember what we did on Google Scholar, we confined it to 2015 and more recent. So we can do the same thing here on Web of Science by checking these boxes and then click refine. And that takes it down to 374. Okay, so now I think that's enough, or small enough, that I can start reading titles and looking for specific journals, articles to start with. But if I wanted to refine this down further, I could do that by scrolling down here, and I could select document types, for example. So I open that, and I can say, okay, I only want review articles or articles. So I don't want patents, others, or meetings. So we're going to confine it to articles and review articles. And that should give us about 200 results, 191. Okay, excellent. So the other thing you'll notice is that these are automatically sorted with the newest on top. Okay, so the newest articles are at the beginning, as opposed to Google Scholar, where it automatically had the most cited articles at the beginning. That's usually how it sorts. But we can change that to time cited. And so now we have the same number of articles, but they're sorted by citation number. And you can see the citation number over here. This article's been cited 94 times since 2015. But I don't see earthworm or plant or root or growth in the title. So the keywords are going to be highlighted in the title. So if we go down here, we can see growth, plant, here's earthworm, growth, plant. Okay, so none of the titles are really showing all 4 of those key terms yet. So I'm going to keep searching, but first what I'm going to do is have it show 50 articles per page. That way I don't have to click through pages as many times. And so I'm just going to pick one at random here. So we can do some of the same things we did with Google Scholar. We can click on articles that have cited this one. So if I click on the 17, that shows all the articles that have cited this particular article. If I click on the title, it's going to take me to another page where I can read the abstract. Okay, and I can also click this 55 and see all of the papers that were references in this particular paper. So I can search backwards in time by doing that. Then I can search forward in time by clicking all of the articles that have cited this one, the 16 here. It was 17 on the other page, but it's showing 16 here. Doesn't matter. That's what you do with that number. And then again, we could click any of the authors. But you can do that. You can click on author names, and just like you did in Google Scholar. The other thing you can do similarly to Google Scholar is click View Related Records. And this will give you a long list of related publications. Okay, so it gave us a list of almost 7,000 that are related to this paper. So that's a bit surprising. Some other ways you can refine these long lists is, let's say you want to see only highly cited papers. You can click that button. Or only hot papers in the field. So if we refine it to highly cited and hot papers, that should take it down. What's really nice about Web of Science here is we can take a lot of articles at one time. So let's say I want to put everything on this page, all 50 of them, into my reference library. Do I have to do it one by one? Not with Web of Science. So I can click this, select page, and now it has selected every article on this page. These are all highly cited. Then you don't have to select everyone. You can go in and select them one by one with this checkbox. Once you've selected all the references you want on the page, click right here, Add to Marked List. Okay, so when we do that, it's going to put them into what's called the marked list. Okay, so we can then go to the second page, and let me just choose 2 or 3 more. Okay, so we want that one and that one. I'm going to add those to my marked list. So now we'll have 52. Let's say I'm done for the day. I want to import all of these into my reference library, EndNote or Reference Manager or RefWorks, whatever you are using. Then I'll click on Marked List, and that will open up the list, and I can output all of these records. So right here it says Output Records. So we can do all records in this list up to 500. So if you have more than 500, then you have to figure out a different way to export them. And then you can choose what kinds of data are going to go out. So you see that down here we have authors, editors, ISSN, ISBN, PubMed ID, title, etc. are already checked. So those are the standard data fields that will go into your Reference Manager database. But if you want more fields, like if you want the abstract to come in there, you can click Abstract. If you want to know the cited reference count, okay, this is one of your homework assignments. If you want to know the cited reference count, you can click that. Anyway, when you've chosen all the fields you want to go in, now go up here and click Export, and then it's going to ask you which one of these to go into. Okay, so if we're going to EndNote Desktop, then I would choose that. If we were going to go into RefWorks, then I would choose RefWorks. Okay, so there's a few other formats here. You can print the list if that's helpful to you. And you could say other file formats. So let's see what happens there. Other reference software. Now we have a choice of BibTeX and plain text, tab delimited, so on and so forth. Okay, so not a lot of choices, but we do have BibTeX, RefWorks, and EndNote. Okay, so once you get them into your reference manager, then it's the same as what we looked at in the other lesson in terms of plugging them into Microsoft Word and into your paper that you're writing. Okay, I hope that helps. Thank you, and I'll talk to you later. All right, welcome students. Today I'm going to do a demonstration to show you how to use Google Scholar to find papers that you're going to read that will become the sources that you cite in the paper that you are going to write. You can look for sources in a lot of different places, like we discussed in the other lessons. You have Web of Science, Scopus, PubMed, just to name a few, but I'm going to show you Google Scholar. And I'm going to show you how Google Scholar interfaces with a reference managing software called EndNote, and I'm going to show you how EndNote, reference managing software, interfaces with Microsoft Word. So if you use these three tools together, it's going to save you a lot of time up front, and it's going to save you a lot of time formatting your references, and it's going to save you a ton of time if you have to reformat your references later, like if you change target journals or if you have to resubmit to a different journal. What we're going to do first is we're going to search for a topic. So let's say you're just starting in a field and you don't know much about this field. So this would be the case for me. In this topic, and I want to know the effect of earthworms on plant root growth. I'm just going to type those terms, earthworm, plant root growth, hit return, and so, well, that was pretty easy. Actually, we got 46,900 results, but the first one is basically right on topic. Okay, it doesn't usually happen that easily, but there you go. Okay. The problem is that this article is a bit old. It's from 2003, and currently we're at 2019 or 2020. So that's a long time ago. I want to restrict my search to newer articles, and also I don't want to have to look through 47,000 titles. Okay, so I want to tune this down to a smaller amount. So the first thing I'll do is unclick this include citations. Okay, that took it down by 700. It's still over 46,000 results though, so that's too many. I'm going to only consider papers that were published in 2015 or later at this point. Now, once we find a few key articles, I'll show you how Google Scholar can allow you to expand your search older than that and also newer than that. So restricting it right now is not going to cause you to miss anything once you do your extended search. So I'll just click since 2015. That brings it down to 16,000 results. Okay, that's still a lot to look through. But just to keep the demonstration moving, what we're going to do is say, well, I really like root traits and root biomass allocation impact how wheat genotypes respond to organic amendments and earthworms. Okay, so there's a lot going on there. We've got the genetic component of the wheat plants and how that affects the way they grow and how that affects the way they respond to earthworms. So that could be a good starting point. That was published in 2018. And so what I'm going to do is say I think I want to look at that one more closely. So the first thing I want to do, if I'm interested in that paper, is to put it into my reference library. So I want to build up my reference library as I'm doing the search process. So I'm just going to grab everything that looks like it's interesting. If I don't cite it later, I can delete it from the library or just leave it there. It won't hurt anything to leave it there. So the way to put that into your library is to go here, these little quotation marks, and click on that. So when we click that, this window pops open, and you see it's got the reference for this paper in a few different styles where you can click and copy and paste those. But at the bottom, you see some hyperlinks here where you can dump this reference into some different reference managing software. So BibTeX, EndNote, RefMan, and RefWorks. Today we're going to be looking at EndNote. So all you have to do is click on EndNote, and it's going to download that reference. I'll take it from my downloads, drop it into EndNote, and there it is. So here's the reference. This is the start of my reference library, so I can click on that. And what we see is it's imported all of the authors, the year, the title, the source, so the journal it came from, the volume, the issue, the page numbers. And down here in this window, you can see what the reference is going to look like when you put it in your paper. All right, so what I'm going to do now is switch back over to Google Scholar. You can close this. And so this is the paper we just looked at. Now, if you look over to the right, it says html at plos.org. So if I want to read that paper, I can click this link right here, and that is going to go to PLOS One journal. And there it is. So I can download the PDF by clicking this, or if I go over to this little down arrow, I can also get the citation here. So if I didn't want to pull it out of Google Scholar, I could pull it from the journal's website also. Now, this is the paper we've been dealing with. All right, so if that's our starting point, now what we can also do is look at papers that have cited this paper. And At the time I'm recording this, it's still pretty recent, so it's only been cited twice. But if I click on that, I'm going to do it by opening a new tab. So I open that in a new tab, switch over to it, and we can see that the two papers have cited that. And if I read the titles of those, I can see one of them is more about the wheat varieties than it is about earthworms. And the other one is looks like it's more about root functional traits, not so much about earthworms. Okay, so this time the papers that cited that one kind of struck out. But usually that will give you some good results. The other thing you can do here is click on related articles. Again, I'll open that in a different tab. And so you can see we got 101 of those related articles. So you could scroll through here, and just glance at the titles. But again, a lot of times that will give you some more leads onto important related articles. So I'll close that tab. Now we're back to this article again. The other thing you can do here is you see some of the names of the authors are underlined. And so if they're underlined, then those are links. So if we go to the last author, usually in this field, the last author is usually the professor or the leader of the group. So if we click the name, we'll see all the other articles that Stephen Fonte has published. So here we go, earthworm populations in California tomato cropping systems. Okay, that looks pretty good. So, and we can get the PDF of this paper by clicking here and it'll take us to ResearchGate. Or if we click the title, that's going to take us directly to the journal. So if you're a university student or postdoc and you're on campus, you probably your library has access to this. And let's choose this one from 2003 and just look at the articles that have cited that one, because that one's really exactly what I was looking for. Effects of earthworms on plant growth. So 390 papers have cited this one since 2003. I'll click on that. And the other thing you can do when you go into it that way is you can search within the citing articles. So now let's say I want to search only for ones that do have this word, earthworm or earthworms. And most of them do. 354 of them do. Okay, so I'm just going to cite a few of these. All right, so we have 5 references. So what I'm going to do now is, let's just assume that this is 200 references that you've built up. So you're going to keep doing this search process, building your reference library, collecting the papers, the PDFs, and then later you'll go and sit down and read all those papers. You'll spend a few hours reading, and then you'll make notes on them and decide which ones are the important ones that you're going to cite. So what we're going to do now is switch over to Microsoft Word, and we're going to start typing our paper. Okay, so let's assume you've written an entire paragraph here. And so as you're writing these sentences, you want to cite references. So earthworms live in the soil. And I'm going to put a space there because that's where I'm going to drop in the citations. So I will switch back to EndNote. So you click one, and then if you need to select 2 or 3 references, you can hold down the command button and select other ones that are not necessarily next to each other. So I've selected 2 references. Now I'll go back to Word, and I've already installed when I installed EndNote on my computer, I also installed the add-in into Microsoft Word. So now I can go here to EndNote X9, click that, and then I go over to Insert Citation, and then I pull down the menu, Insert Selected Citations. So I click that, and then instantly what happens is the 2 citations go in, and it starts to build my bibliography. Yeah, so that's pretty awesome. I didn't have to type any of that. Now, the second sentence, we say they are beneficial to plants. So we're going to put some citations there. Switch back to EndNote. And I'm going to select the other 3. Switch back to Word. Insert selected citations. Okay, so now it has 1 and 2, 3 through 5, and all 5 of them are showing up here. All right, that's awesome. So now I can put my references subheading there and just keep going. Just keep writing and citing, writing and citing. Now, one thing that we're going to notice is this is just a style that's putting the references as number 1 and number 2, or 3 through 5. So let's say your target journal doesn't do it by number, it does it by name. Okay, so we're going to need to change that. So what you can do is go up here, and it's set for numbered, and what we're going to do is select a different style. Okay, so I'm going to say select another style. All right, and when you do that, it pops up this window. Sometimes your journal is not on the list, and you'll have to figure out how to add it to the list. That's not that hard to do. You just have to Google and find out how to do it. Scroll down here and find planta. And I choose that. Okay, so now it goes into the planta format, and it has the names and the years instead of just numbers. And you see it's completely reformatted all of the big bibliography. So another thing you need to do is go through the bibliography and make sure it matches with the instructions for your target journal. So I can see at least one problem right now, and that's right here in the title of plant disease. It didn't capitalize the D in disease. It should be capitalized. So I could fix it here in Word, but that's not a permanent fix, because when I update this, it goes right back to the lowercase. Why does it do that? It does that because I didn't update my source material, my EndNote library. So I have to go into EndNote, and so I'm going to open that reference, and then I'm going to type and correct that typo. All right. And then I close it and save it. And then if I go back to Word and I update, then it's correct. Okay, plant disease is now correct. So this is one of the drawbacks of using Google Scholar. It tends to not always capitalize all the words of the titles. And so you have to do corrections for that. The other thing to look for is italics on the species names. A lot of times when you copy these references over, the italics don't come over with it, and you have to go into EndNote and correct that. So that's true with any of these reference managing software packages. It's not an EndNote specific thing. Okay, well, I hope that's helpful. So let's review what we looked at today. We looked at how to go into Google Scholar and find source material, get the PDFs of those papers in many cases. how to import the reference into the reference library, how to start with that one reference and expand the search in different directions, past and related articles and by author. And we also looked at how to import the references into EndNote, how to take the references from EndNote and put them into Microsoft Word, build the bibliography, and change the target journal format when necessary. So now it's time for you to put this into practice, and it does take practice. I went through this very quickly, but you can watch the video several times and go out there and start finding your source material for your paper. Good luck. Hello and welcome to Lesson 3. In this lesson, we're going to give an overview of the writing process. We'll talk about pre-writing. We'll talk about what happens when you submit your manuscript. And we will talk about some tips on how to write your first draft. To begin, let's take a look at where we're at with the overall project management approach and look at the graphic. So for this lesson, we are here in the planning stage. In a previous lesson, we talked about scientific paper structure. That was lesson 1. In lesson 2, we talked about finding your references and building up your reference library in the planning stage. And now we're talking about pre-writing here in the planning stage. The learning outcomes for this lesson are as follows. When you conclude this lesson, you should be able to explain the submission and review process. You should be able to identify a single target journal. I want you to be able to determine who should be authors on your paper, and as part of that process, who should not be authors on your paper, but should be in the acknowledgments section. And fourthly, at the end of this lesson, you will plan your first draft, you'll make a writing plan, and you'll have a rough outline of your article. So let's just take a step back and think about the entire process of writing a scientific paper. We can look at it as a project management task, and we can also think about the writing process all the way up to submission as occurring in a few different stages. And I've listed 6 stages here. What we're talking about here is breaking down the writing process into planning, pre-writing, drafting, revising, editing, and submitting. So today we are talking about planning and pre-writing. So in the planning part of writing a paper, which is also in the planning part of the project management approach, first think about our intended audience. That is, who do you want to read your paper? So think very carefully about the characteristics of that audience. Which journals do they read? What types of techniques are they interested in? What types of questions are they asking in their research? What kind of results do they need from you to advance their research? So think very carefully about exactly who you want to read your paper. And that's going to help you narrow down your list of target journals. And then you're also going to make a writing plan. What you're going to do is collect and review literature, annotate that literature, and organize it. Now, you should be able to do that with what we talked about in our first 2 lessons. So we talked about how to read scientific papers, how they're structured, what to look for in terms of key elements. And in lesson 2, we talked about finding literature and collecting it, building up your database of references. So this is going to match well with what we talked about before. In the drafting stage of writing your paper, what you'll do is write all the sections of the paper. And then after you have this complete first draft, what you'll do is start with revising and editing. Now you are ready to submit your paper to your target journal. where it will go through the peer review process. Let's talk about what happens when you submit your paper to a journal for review. Now, this is something that I didn't understand for a long time. I'd actually published quite a few papers, and I was a corresponding author before I got a good handle on this scientific review process. So the first step is that the corresponding author will submit the manuscript and all the figures and supplementary files through the journal's website. And once it's submitted, it will go to an associate editor. Now, different journals may have different names for this position, but a lot of journals call it associate editor, so we'll just go with that. Now, the associate editor will look over your paper. He or she will read your cover letter, and he or she will read your abstract and glance through the figures and tables and think about whether it's a good fit for this particular journal. Does it meet the needs of the audience? Does it match the scope and the aims of the journal? Is it a complete paper? Does it look like it was done in a scientifically sound manner? And at this point, they may decide to reject the paper. This is called a desk reject. And the desk reject usually happens in between one day and 1 week. It's usually pretty quick. And so reasons you might get a desk reject are because the subject matter of your article doesn't match well with the journal. If you're trying for a high impact journal, the associate editor may feel like the work is not novel enough or will not have enough of an impact, and they might tell you to submit it somewhere else. Other reasons for a desk reject are technical reasons. You didn't follow some of the instructions, in which case you might be able to correct those mistakes and then resubmit. If the associate editor decides to send your paper into the review process, then they're going to send it to a handling editor. And that handling editor is then going to recruit peer reviewers, and they'll recruit 2, sometimes more peer reviewers who will carefully read your paper. They'll evaluate its strengths, its weaknesses, the scientific rigor of the methods and of the results and the interpretation, and they'll make a recommendation, and that recommendation will go back to the handling editor. So once the recommendation goes back to the handling editor, that editor will make a decision. And at the same time they make the decision, they will send the peer reviewers' reports back to the corresponding author. Let's look at the decisions that could be made. So the decision could be a hard reject. In other words, the journal is not going to publish your work, even if you revise it and resubmit it. They just don't want to publish it, and they'll tell you that you should try to publish it somewhere else. The next decision that could be made is a reject and resubmit decision. So the paper is not going to be accepted in its current form, but if you make major revisions, they would be willing to take another look at your paper. You could resubmit it and potentially get a better decision next time. That's a very common outcome. It's probably the most common outcome, is either an outright rejection or reject and resubmit. When one of your papers gets rejected, it's going to hurt, but don't feel too bad because that is the most common situation. Now, a third decision that could happen is you might get what's called a minor revisions, or some journals say could be accepted with revisions. That is really the best decision that you can hope for. That's the best decision you will get. The fourth option is accept. And that's not going to happen the first time. Now, if you get one of these decisions that allows you to resubmit, so a reject and resubmit, or it could be accepted with revisions, now's your chance to address the peer reviewers' reports. So those reports came back to the corresponding author. That author will share it with the rest of the co-authors. And what you will do is when you resubmit the paper, you will go through all of the reviewer comments and address them point by point, while at the same time revising your manuscript to take those reviewer comments into account. Now, there may be times when you think the reviewer comments are incorrect or not applicable, and you don't want to do what they're telling you you should do. In that case, you need to make a strong scientific justification for not following the recommended changes. Otherwise, if you agree with what the reviewer is saying, then go ahead and make those changes and indicate what you did in your response. And usually the reviewers pick up on some things that you missed, and they do a good job at helping you make your paper better. Now, after you resubmit, it will go back through this process. It'll go back to the handling editor. If it was a reject and resubmit, it will definitely go back to the reviewers. If it's a minor revisions decision, it may or may not go back to the reviewers. And then at that point, you could get an accept decision, or you might get another minor revisions decision. So you might have to go through revisions 2 times or 3 times, maybe even more, before you get to that accept decision. But once you get to accept, it's going to be worth it, because that's just such a great feeling when your paper is accepted for publication. Now, why did I tell you that you're not going to get an accept decision on the first submission? Let me show you this quote from Joshua Schimmel. He's the author of a book called Writing Science. He says, "I've authored 175 papers, written hundreds of reviews, and handled about 2,000 manuscripts as an editor. Across all those, not a single paper has ever been accepted outright. Not one. Some only needed a light bandage, others required major surgery, but they all needed some editorial care." And so I think those numbers will tell you that the chances of your paper being accepted on the first try are very close to 0. And I'm not telling you that to make you feel anxious or apprehensive. I'm just telling you that so you'll know what to expect. Now let's talk some more about your target journal. How do you find your target journal? So first we talked about the audience, and that's a big part of it. So figure out what audience you want to reach, and then figure out which journals that audience reads. One thing you can do is look for papers that are on a similar topic and see where they were published. So that list of journals is going to be a good possibility for your target journal. When you want to see the audience of the journal, what you can do is go to the journal's website, where it talks about the scope of the journal and the types of papers that they're looking for, the types of techniques they're looking for, and the types of topics that they're looking for. And that'll give you some ideas about where to submit it. Of course, talk with all your co-authors as you're working towards finding a target journal, and everyone should be in agreement on where to submit it. So get some input from your co-authors as well. Once you decide on your target journal, before you start writing, now is the time to download or print out the instructions for authors. Follow those instructions very closely. These instructions are going to tell you how to write the paper in terms of what should be on the title page, what types of keywords and how many keywords you need, if you need keywords, the length and the format of the abstract, so the word limit for abstract, if there are word limits on the manuscript itself, and it will tell you what kinds of file formats you can upload to the website, both for the text file and for your figure files and your supplementary files. It's also going to tell you how to format your references. And it's a very good idea to cross-reference the journal's instructions with the output in your reference managing software. They should match up, but sometimes they don't, and you have to edit the output style. I've had to do that a few times. And if you decide after you've written the paper that you need to submit it to a different journal, then you can reformat all your references in about 5 seconds compared to retyping all of them, which will take you probably 2 or 3 full days of work. So it's time to talk about authorship. As you know, in science, authorship is really important. So authorship is how you build your CV. It's how you get your next job. It's how you get promoted. It shows your record, your productivity. So being an author on a scientific paper is a really big deal. It's such a big deal that it's a leading source of conflicts in the academic world. And so that's why it's really important to start talking about authorship now, before you start writing your paper. What you should do is have everyone who has been involved in the project in on a meeting or an email chain, and everyone needs to contribute and agree on who the authors are going to be and in what order they're going to be listed. The order of the authors is also important in most scientific fields. So decide on this before you start writing. Now, who should not be an author? Just because somebody worked on the project doesn't mean they automatically earn authorship. They need to have significant intellectual contributions to the work to become an author. Now, let me give you an example. Maybe you hired a research student who you're paying by the hour, and they're collecting a lot of data for you and putting it into an Excel sheet or writing it down in a notebook. Does that student worker earn authorship on the paper? No, they do not, unless they have contributed something more than just collecting data. One thing that's important is that all of the co-authors are going to read and approve the final version of the manuscript. One thing you should do is look at your target journal and see if they have guidance on authorship. Now we're going to look at some guidance for authorship now. This is put out from the International Committee of Medical Journal Editors, who publishes the uniform requirements for manuscripts submitted to biomedical journals. Now these uniform requirements can be applied to other disciplines. They don't have to only be for biomedical journals. So let's take a close look at this authorship statement. Authorship should be based on the following 4 criteria. Point number 1, substantial contributions to the conception or design of the work, or the acquisition, analysis, or interpretation of data for the work. And number 2, drafting the work or revising it critically for important intellectual content. And point number 3, final approval of the version to be published. And point number 4, agreement to be accountable for all aspects of the work in ensuring that questions related to the accuracy or integrity of any part of the work are appropriately investigated and resolved. In other words, if there's someone on the team who did plagiarism or fabrication or falsification of some of the data, everyone on the team is responsible for that. You can't just blame that other person and say it's not my fault. Everyone is responsible. Let's continue reading this statement. In addition to being accountable for the parts of the work he or she has done, an author should be able to identify which co-authors are responsible for specific other parts of the work. In addition, authors should have confidence in the integrity of the contributions of their co-authors. Now, some journals ask the authors to make a statement of contributions of the individual contributors. And that statement is usually somewhere near the acknowledgments at the end of the article. The final sentence of the statement is, all those designated as authors should meet all 4 criteria for authorship, and all who meet the 4 criteria should be identified as authors. Those who do not meet all 4 criteria should be acknowledged. In summary, these authorship rules are quite strict, and if you want to follow ethical publication practices, you should pay close attention to these guidelines. The order of the author names is also quite important for journal articles. And I should point out that different fields have different conventions. For example, some fields in science list all the authors alphabetically, regardless of who contributed the most, who contributed the least, and who is the head of the laboratory. But for most of the life sciences, these are the typical author order conventions. The first author is the person who contributed the most to the research, and usually they're also the lead author. They did most of the writing. The last author is typically the head of the laboratory, And the corresponding author is usually the first author or the last author. Now, sometimes there are co-first authors. This is when two people made an equal contribution to the work that's being published. And so there's typically an asterisk by their name stating that these two people contributed equally. However, on the piece of paper, somebody's name is going to be first. And so it's important that you talk about whose name is going to be listed first, even in the case when there are co-first authors. Now I would like to give you some tips for writing the first draft. The first tip I have is to organize your results and your ideas by making an outline. So you can do this using the reader-writer template that I have provided to you in this course. You could do something like draw storyboards on a whiteboard. You could draw on paper. You could make a traditional outline like you see on the bottom left part of the screen that we talked about earlier. Or you could write your key points on post-it notes and move those around on a wall or on a table, or you can use index cards. Whatever works for you to think through how you're going to organize your paper. But it's going to help you tremendously to have a writing plan at this point and have an outline to get started. Now, you may adjust your outline later as we get into each of the specific sections, and that's okay, but at least have something to get started with. Some additional tips for writing the first draft. A good place to start is with the research question, and then work backwards from the research question to determine exactly which background information you need to provide in the introduction to funnel down to those knowledge gaps that will reveal your research question. From the research question, you will also work forward to the results and forward to the discussion section. Now I have some additional tips for writing the first draft. These are tips about writing process and writing productivity. So when you're writing the first draft, the main thing is to get the words down. So don't worry about the length. It might be too long on the first draft or it might be too short on the first draft. Either one is okay. The second tip is don't edit as you write. This is going to slow you down tremendously, and it might be a total waste of time because if you spend 10 or 15 minutes getting a sentence perfect and then in revisions you delete that sentence entirely, well, you can see that you wasted all that time editing. Even if you know that the sentence is not very good or you feel like the sentence is not very good, keep moving. Just get the words down. I also recommend that you plug in the references as you go. That's going to make it a lot easier later. But if you find that plugging in the references as you go is slowing you down too much, you can leave a placeholder there to come back and put in the reference later. Now, one important thing is if you get stuck, like if you're starting to write and you just can't think of the next thing to put down, don't stay stuck and keep working on that same spot. What you should do is just write yourself a placeholder note saying, come back to this point and fill in this concept, and then keep going. You just want to get the words down on paper. Remember, you can't edit an empty page. Some more productivity tips for you on writing the first draft. Make sure that you write when you have a few spare minutes. So if you have 10 minutes between classes, if you have 10 minutes or 15 minutes between meetings, you can get a lot of work done in that small amount of time. All you have to do is sit down and start writing. Don't wait until you have the 3 or 4 hour block of time, because that's not going to happen very often. So it's really important that you be able to start writing on command. And you need to write even when the conditions are not ideal. Also, you need to be able to write consistently. I suggest every day and writing for 30 minutes every day. Now, whether every day for you includes the weekends or not, that's your choice. But at least during the week, write for 30 minutes every day. Make that a key priority. My point about writing every day is really about consistency. It's much better to be consistently writing every day than to be writing one day a week for a huge amount of time. So make sure you write even when you don't feel like it. As scientists, we are also writers because our work is not done until it's written and published. And so we have to be able to work on command. Writing is part of our job. It's not something to do when we feel like it or when we're inspired to write. We need to write when we need to write, no matter what's going on, even if you lost your lucky pencil. You are a writer, and what do writers do? They write. Now, I'd like to show you a couple of quotes. Harlan Ellison says that people on the outside think there's something magical about writing, that you go up in the attic at midnight and cast the bones and come down in the morning with the story. But it isn't like that. You sit in the back of the typewriter and you work, and that's all there is to it. I don't think any of us are going to be using typewriters, but you sit in front of your laptop or your desktop and you work, and that's all there is to it. Here's another quote from Ann Tyler. "If I waited till I felt like writing, I'd never write at all." I know what she means, but I'll tell you, as someone who's been a full-time writer, when you get into the habit of consistently sitting down and writing, it becomes easier. Do you have writer's block? Can you have writer's block as a scientific writer? Well, I believe you can't. Scientific writers don't get writer's block because we're not creating stories out of the thin air. We're not writing novels. We're not writing fiction and creative stories. We are documenting and we are explaining and we are interpreting. We're not afflicted with this thing called writer's block. If you're having trouble getting started with your writing, that's not writer's block. That is procrastination. I really think this quote from Steve Martin is funny. Writer's block is a fancy term made up by whiners so they can have an excuse to drink alcohol. So even if you don't drink alcohol, that's a funny quote. All right, we are to our action list for the week. First, I want you to call a meeting with all of your co-authors and determine who is going to be an author on your paper and determine the order that the authors will appear on the author list. This is the only thing on your action list that I recognize you might not be able to finish before you move on to the next lesson. But get it started at this point. Secondly, decide on a target journal and get those instructions to authors and start reading them and familiarizing yourself with the format that journal wants you to submit your paper. The third and fourth items here we didn't talk about during this lesson, but I want you to go ahead and get started on analyzing your data and sketching out your figures and tables. So even if you don't have all of the final data yet, if you're still collecting some data and analyzing it, you still know roughly what your figures and tables are going to look like, or at least what results they will contain. You may not know how those results are going to turn out yet, but you know what's going to be in those figures and tables. So sketch those out and then put all this together and start forming an outline for your paper using the reader-writer form that I have provided you or some other method that we talked about. As we wrap up, I would just like to reiterate the learning outcomes for this lesson. Explain the submission and review process, identify a target journal, determine who should and should not be authors, and plan your first draft and outline your article. Again, please don't move on to the next lesson until you have achieved all of these learning outcomes. Go ahead and get to work, and happy writing. Hello, welcome to the lesson on figures and tables. Let's take a look at where this lesson fits into our project management approach of writing your manuscript. We are still in the planning phase. I would consider making figures and tables part of the pre-writing process. So this lesson on figures and tables will conclude the planning phase. Then we'll be ready to move on into executing phase. This is one of the shorter lessons. However, that doesn't mean that there's less to do. It depends on how much you have already done with your figures and tables, and how many figures and tables you have and how complicated they are. So although it's going to be a short video, it's still going to be a lot of work for you this week. The learning outcomes are as such. At the conclusion of this lesson, you should be able to determine when to use a table, when to use a figure, or when to use a photograph or a diagram or some other kind of figure. And you should be able to explain the concepts for figure design and presentation. Now, why do we have figures and tables? Let's step back and think about that. Figures and tables are how you present your processed data. So you collected raw data, and then you processed it by analyzing it with whatever method is appropriate for your field and for the type of experiments you did. And now in the figures and tables, you're going to present that processed data. So it might be means, plus or minus standard deviations. In some cases, you might be presenting the raw data that has a curve fit to it. And there's a lot of different ways that it can be presented. The whole point of figures and tables is to show the readers the outcomes of your experiments that you're doing to fill the knowledge gap and answer the research question. So how do you decide how to present your results? Are you going to use a figure? Are you going to use a table? Or maybe you'll just put it into the text. When you have only a few data points, let's say 4 or fewer, it doesn't make a lot of sense to make a table or a figure for that small amount of data. And so in that case, it's best to just put it into the text in a sentence in your results paragraph. A figure is going to be the best choice most of the time because figures are a lot more visual and they're easier to understand quickly, and they allow you to see trends and relationships more easily than a table does, where a table has a lot of numbers and it's more difficult to read. If you can use a figure, do that. When you need to use a table would be when you have a mixture of data types that would take too many figures to present, and so maybe it's more compact in a table. Another time you might want to use a table is when you need your readers to see precise numbers. So if the exact number is more important than the trends, for example, then a table would be the best choice. So let's talk a little bit about table design. When you design your tables, keep the reader in mind. This is always going to be something we come back to in this course, is keeping the reader in mind. What you want to do is make the table easy for the reader to follow. One way to do that is to keep the structure as simple as possible. Additionally, you should place familiar information on the left and new and important information on the right. For example, times or dates would be in the left-hand column, and then the numbers that you collected at those times or dates would be in the right side, to the right side of that first column. Also think about the table titles. So these are kind of equivalent to the captions for figures, but table titles are a little bit different because you don't have an extensive legend for tables. You have basically a descriptive title. Additionally, think about what you're going to put in the columns and what you're going to put in the rows. So you have headings for the columns and you have headings for the rows. It's easier for readers if you put the dependent variables in the columns and the independent variables in the rows. I think this goes along with placing the familiar information on the left. Now, when we think about figures, there are a lot of different types of figures, and I feel like it's out of the scope of this course to go into a lot of details about types of figures and how to make those types of figures. So I'd like to just focus on an awareness of them, and then I think you should get advice from your colleagues and senior scientists in your group about specific figures that you need for your paper. But in general, the different types of figures that you might choose to use are shown in this slide. So there are photographs. Use those when you need to show specific samples, microscopic images, aerial images, kinds of things that you can only capture in an image. They're very specific to your study. Drawings and diagrams can be very useful when you need to show flowcharts or decision trees, show how you built something or how a pipeline was constructed. Drawings and diagrams are great for that. Line graphs are probably the most common type of figure, and they're used to show dynamic relationships over time, concentrations over temperatures, different changing variables that might affect whatever is being measured. So that's what I mean by dynamic relationships. Scatter plots are great when you need to show relationships between two variables. So maybe you're measuring height versus weight, and you would have one data point for each height versus weight relationship for all of the subjects that you're measuring. And so you could plot that as a scatter plot. And you might fit a curve to that, or you might not. Bar graphs are great when you have groupings of treatments, if you have different genotypes that you're comparing, or those kinds of situations. Pie charts are used occasionally. I don't see them used a lot, but they are handy when you're dividing the whole group of results into percentage categories. Box plots are great. They're kind of like a bar graph, but they have a lot more information in them, and they show the variation, and they show variation that can't be captured in traditional bar graphs, because not only do they show the mean, they also show the range, and they show outliers. They have the whiskers that extend beyond the boxes that have a lot of additional information. And then there are also principal components, figures that show how certain treatments or genotypes cluster together. So these are just some examples of figure types that you might use. You might use something that's not even on this chart. As you are making your figures or tables, keeping the reader in mind, I think it's important that you think about the following points. What is the primary outcome, in other words, the result, from this figure or table? So if you're going to make a figure and you're thinking about which type of figure to make, it's useful to look at this question and think about why you did that experiment, why you collected that data. And that's going to help you decide what's the best way to present it. Secondly, after you've made your figure or table, or while you're making your figure and table, think about if you're presenting the data in a way that allows the reader to quickly see the results. So in other words, are you presenting the data in a way that makes it easy for the reader? Now at the same time, you don't want to cross the line to where you're leading the reader to a certain conclusion. So think about this question: is my presentation unbiased? Make sure your presentation is unbiased and it is not misleading. We're not setting out to reach a foregone conclusion. We're trying to find out the truth about nature and natural phenomenon. So we need to present the data in a non-biased way, and at the same time in a way that it's easy for the reader to see the outcome of the experiments. So for example, if you're making a bar graph or a line graph and you're comparing two different treatments, think about where you start and stop the scale. So should you start the scale at 0? Let's say if your numbers are 900 and 950, should you start the scale at 0 or should you start the scale at 850? If you present it as 850 to 1000, that could be misleading because it's going to make the distance between those two lines much greater than they would be if you started the scale at 0 and went up to 1000. So think very carefully about how you design the figures for that purpose. What kind of software do people use to make figures or tables? Excel is used quite frequently. Now, I would not recommend that you output the default Excel graphs. That looks pretty amateur. If you do use Excel, then you need to make a lot of changes so that they look professional and not just quickly slapped together. A lot of people will export their figures into Photoshop or Illustrator to clean them up and make them look better. ImageJ is a software that's similar to Photoshop, but it's a free version provided by the NIH. Sigmaplot is commonly used, Cricut Graph. A lot of people are using R these days to make figures, and R is a pretty powerful program that makes really nice looking visuals. And there are probably other ways to do it that I haven't mentioned here. So going back to this idea of readability and making the reader's job easy, let's look at these points. First, make sure you're using large enough lettering and large enough symbols that when your figure is printed, the size that it will be printed in the final journal article, that it's still easy to read. A common mistake I see is that people are using too small of a font size or too small symbols. I don't want to pull out a magnifying glass to be able to read the figures. Also, with symbols, it's not just about size, but it's also about distinguishing them. So it can be hard to tell a diamond from a circle sometimes, especially on certain types of graphs, like scatter plots. Be aware of overlapping lines or curves. If you're making line graphs, if the curves are overlapping each other, it can be really hard for the eye to trace which curve is which. And if you have too many lines or curves in the graph, it makes it crowded. Consider breaking that figure into multiple panels. If you're going to use fill patterns or shading or colors, do that effectively and in a subtle way. Don't use colors that are harsh and hard to view. Choose colors that work well together. And if you're using shading, make sure you can distinguish the shades. If you're using multiple lines on a line graph, consider using different line weights and line patterns. That makes it easier to follow with the eye. And if you must use color, make sure that you're using color in a way that information is not lost if your readers print your paper in black and white. And also make sure you include statistical analysis information on the figure using symbols or letters or whatever is appropriate for that type of graph and in your field. Let's look at a couple of examples of figures that I think could have been done better. So this first one is a scatter plot, and it has a curve fit on it, which is fine. The problem is that they have 4 different data sets plotted together, and the only way to distinguish the data points in each data set is by color. And the symbols are exactly the same. So if you print this figure in black and white, all that information is going to disappear. You're just going to see a lot of dots. What the authors could have done is, made the red dots diamonds and made the green dots squares and made the blue dots triangles. Different symbols could be potentially distinguished. Additionally, green and red are not ideal colors to use because there is a substantial percentage of people in the population that are red-green colorblind. Here's another example of a figure that I think could have been done better. So what we see here is on the left side a color version of this figure, and on the right side I've printed the same figure in black and white. This is a figure that shows a bar graph, and I think the layout of the bar graph looks fine. They've got groupings of different genotypes that have been harvested at different times during the year, and they're comparing the yields. What I think could have been done better is with the color and the fill patterns. When we look at the color version, to me, the colors that they chose are just not very pleasant to look at. They kind of hurt my eyes. The orange, the yellow, and the black, their contrast is very harsh, and then there are some that are not even colored. If you're going to use colors, I feel like it's better to use a set of colors that go together, that designers have determined are pleasing to the eye together. And then when we print the figure in black and white, we lose a lot of information. And now some of the bars colors are indistinguishable. None of these bars have any fill patterns. So if they had used fill patterns, then when we go to black and white, we wouldn't have lost that information. All right, it is time for your action list for this week. This is a shorter list than in some of the different weeks. However, there's still a lot to do when you make figures and tables. The first thing I want you to do is finish analyzing your data if you haven't done that already, and then determine the best way to present that data. Third, go ahead and make all of your figures and tables. So the last action item is to write down the main take-home message for each figure and table. So what I do is after I make a figure or a table, I print it on a piece of paper, and then I take a pencil or a pen and I write down what I feel like is the key message that I'm going to present in the results section, and I call that the take-home message. So what do you want the readers to walk away from your figure remembering about that figure? What is the result that they should see in that figure? It might be more than one result, but let's start with the most important one and get that written down. Okay, to wrap up the lesson, let's reiterate the learning outcomes for this lesson. At the conclusion of the lesson, make sure you can determine when to use a figure or when to use a table, and which type of figure to use if you're going to use a figure, and be able to explain the concepts of figure design and presentation in terms of readability and making the reader's job easy. All right, that's all I have for you for this week. Go ahead and make sure you accomplish these learning objectives before you move on to the next lesson, and happy writing! Hello, and welcome to the lesson on writing the introduction. Let's take a look at where we are in the project management approach. We have moved through the initiating phase with scientific paper structure. We have moved through the planning phase with building our reference library and pre-writing, making figures and tables. So we are here at the beginning of the executing phase, getting ready to write the introduction. So this is exciting. We're finally getting into the main body of the work. The learning outcomes for this lesson are as follows. First, I want you to be able to explain the purpose of an introduction. You should be able to explain the key elements in an introduction. You should be able to structure your introduction to funnel from general information to specific information. You should be aware of how to signpost all of your key elements in the introduction section, and you should be able to avoid the common problems of the introduction section. So let's look at a quote from William Zinsser. And William Zinsser was an author who wrote a lot in magazines back when magazines were a big thing. A lot of people read them. And he also wrote a book about writing that's a very highly regarded and well-respected book. And so this quote is from that book. He says, "The most important sentence in any article is the first one. If it doesn't induce the reader to proceed to the second sentence, your article is dead. And if the second sentence doesn't induce the reader to continue to the third sentence, it's equally dead. Of such a progression of sentences, each tugging the reader forward until he is hooked, a writer constructs that faithful unit, the lead." So in our case, I think we should substitute the word sentence for paragraph. So let's say in your scientific article, the first paragraph is the most important one, because it's going to get the reader to continue forward. So let's think about what are the functions of the introduction. So that quote gives away some of that. The function overall of the introduction is to get the reader to read the rest of the paper. So that goes along with our first point here, to get the readers interested in what you will be telling them. Not only that, but you're going to provide the context for what you will be telling them. You're also going to use this section to let the reader know why your topic is important. So how does it fit into the bigger picture? How is it significant? What is the scope of this problem? Or is it a very specific problem that's been hard to solve? Also, and this last point is extremely important, it lets the readers know what to expect in the rest of the paper. And we're going to expand on that idea over the next few slides. When you write the introduction, you're going to have a few different tenses, a few different verb tenses. As you're writing this section, you're going to be citing references to establish the who, what, when, where, why, and how context of the paper. So most of this discussion that you're doing in the introduction section is about current understanding and current knowledge gaps, and therefore you should present it in the present tense. So information that has been published we consider to be part of the knowledge base, so it should be presented in the present tense form. But you might want to present some of the introduction in the past tense form, depending on how the results of these previous studies relate to your research question. So if it's something that has been observed, but you're going to call it into question, that's when you would want to present it in the past tense, as if it's not quite certain and your work is going to address that. So there's going to be a mixture of present tense and past tense in the introduction, and maybe in the final paragraph a little bit of future tense. but most of it's going to be present tense. What is the content that you put into the introduction? First of all, you want to try to keep the introduction short. You're not writing a history of your field. You're not writing a review paper here. You're giving background to the research question. So keep it short and make sure you include the following elements. The background information, so these are the knowns. In the first lesson we talked about knowledge bricks to build up walls of knowledge. That's probably the easiest thing to do. Secondly, you're going to discuss the problems and the unknown factors. These are your unknowns or your knowledge gaps. And so it's a little bit harder to expose those, and you do it by showing what's already known so that you can point out what is not known. Third, in the introduction, you need to discuss how the research will address knowledge gaps that you just pointed out. And this is going to be done in the form of your research question or purpose statement. So it could be a hypothesis or a purpose statement. Now, point number 4 is optional, but I like to include it in the introduction, an overview or a preview of the experimental approach and some of your key results. I like to put it here because I feel like it builds interest. When the readers see a little bit about the approach that you're going to take and you give them a taste of what the results are, that makes it more interesting and builds some suspense, so they want to keep reading at that point. Point number 5 is the significance. So you're going to talk about how your research advances knowledge or how your research can be used in the future. Think about writing your paper as if you are a journalist. So these are the journalist's questions. Who, what, when, where, why, and how. Think about if you're presenting all of that information. The who in this case may not refer to people, but you might treat your research subjects as entities, even if they're not. So, for example, if you're studying iron deficiency in plants, you might treat iron and plants as the who part of the equation. So think about the order you present these facts to tell your story. Try to make sure that they're all there. If any of them are missing, that's going to lead to confusion in your readers, and they're going to not follow your paper as well as they could have. Now, this slide should be familiar to you. You have seen it before. This shows how the background knowledge bricks in your introduction should make the knowledge gaps clear. And when those knowledge gaps are clear, that's going to logically lead you directly to a research question that you can pose to address those knowledge gaps and fill them. So the knowledge gaps are going to frame your research question, and then that's going to lead directly to objectives 1, 2, 3, or however many objectives you break that research question into. So again, remember that the introduction has a funnel structure. The first paragraph is going to be the broadest, the most general. And what you're going to do in that first paragraph is to set the stage by giving the broad context of your work. In other words, the 30,000-foot view. So when you're looking out the window of the airplane, you can see the big picture, right? You see the overall landscape. That's what you're doing in this first paragraph. You're trying to also explain the scope and the importance of your topic. So how many people have the disease? How much does it cost because of this problem? Those kinds of things. Numbers are really helpful to explain the scope. As you move into the second paragraph and following that into the middle paragraphs, you're going to give an increasingly more specific background to keep establishing your knowledge bricks or the knowns. And that's going to lead you within those second or third paragraphs to the knowledge gaps. So what is not known in your field. Continuing on with the funnel structure, when you get to the next to last paragraph, this is where you're going to present the research question that, if you answer it, will fill the knowledge gap. And depending on the type of research you're doing, you may present this research question as a purpose statement, or you may present it as a hypothesis. I just like to use the umbrella term research question because it captures any type of scientific setup. Then in the final paragraph of the introduction, what you'll do is briefly describe your experimental approach, give a preview or a teaser of your key results, and talk about the significance again, but in an outward directed sense, showing how your work is going to impact your field and how your results are going to impact society. So we have significance in terms of the scope of the problem, and then we have, at the end of the introduction, significance in terms of the application of your knowledge. So let's go back to this graphic one more time. What I recommend you do is start writing the introduction with the research question, and then work backwards from the research question to the knowledge gaps, and then to the background that you need to show to illustrate those knowledge gaps. And then you also move forward from there. So the research question is really your central theme. Your entire paper is going to be written to reflect back to this research question. You're going to come back to it in the results. You're going to come back to it in the discussion. So set it up very carefully in the introduction. This is going to make your job a lot easier as you write the paper, and it's going to make your reader's job a lot easier if you have written it very carefully. Now, how do you go about thinking about how to structure your introduction? How should I connect all of these ideas? There's a lot going on there, and sometimes it's really hard to figure out how to present it in a way that's clear and easy to follow for the readers. So let's look at this form that's called the message box. Now, this is not something I invented. I'm not sure who invented this, but it's a very useful form, and it has 5 different parts. So you have a central section which is labeled the topic or issue, and then outer sections that are labeled why is it important, what is the problem, what is the solution, and what are the benefits of solving this problem. So the way to use this form is to basically just write out the most simple and concise explanation for each of these prompts. Don't write 3 or 4 sentences here, just write a phrase. You can start in any order. It doesn't really matter which order you fill in the rest of the boxes. Answer what specifically is the problem, explain why this problem is important, explain the solution to the problem, and explain the benefits of solving it. So let's look at an example from my research. So the topic or issue is iron deficiency chlorosis, or IDC, in soybean. Let's start at the top. Why is it important? IDC causes $260 million in losses each year, and new resistant varieties would reduce this loss. What is the problem? Well, we don't know which genes confirm IDC resistance. What is the solution to this problem? In part, the solution could be to measure gene expression in both IDC resistant and susceptible varieties, then determine genes that are highly expressed in the resistant variety. What are the benefits? These highly expressed genes could be new targets for developing IDC-resistant crops. Now, this figure only took me 2 or 3 minutes to explain it to you, but it took me 2 or 3 hours to do this. It's not as easy as it looks. You're going to sit down and spend some time with this form, and it's going to help you figure out how to structure the order of your introduction section. Now, before I told you to keep the introduction short, well, how long should it be? Back in one of your earlier lessons, I had you do an assignment where you went into journal articles in your field and you counted the numbers of paragraphs in each section. So take a look at that. What is the average and what is the range? In my experience, 4 to 6 paragraphs should be plenty long for almost all papers. You want the readers to move through it quickly, get excited, and keep going into the rest of the paper. As you're writing the key elements of your paper, you want to make sure that the readers notice them. Don't hide them, don't make them obscured. So how do you make sure that the readers can see all of these key elements? What you're going to do is signpost them, signal and signpost these key elements. So when you state the unknowns, the knowledge gaps, or problem, do it very clearly and very specifically also. So some key signpost examples that you might see are phrases like has not been determined, has not been established, is unclear, is unknown. Here's an example at the bottom of the slide from a paper that I wrote. It says, however, whether IDC results from low iron supply, alkaline stress, or a combination of these factors is still unclear. One of our objectives was to determine how pH influences physiological and molecular responses to iron deficiency in roots and leaves. So I've underlined the signpost terms. However, signals that there's something coming up that is in conflict. The ending of that sentence is still unclear, is a clear signpost that I have just said something is a knowledge gap. And then I specifically point out the objective that's going to address this knowledge gap. One mistake that authors can make that obscures these key elements is to be indirect in their language. Let's look at an example here from Steven Pinker. He quotes from a paper, "In recent years, an increasing number of psychologists and linguists have turned their attention to the problem of child language acquisition. In this article, recent research on this process will be reviewed." Okay, so that first sentence, if we read it literally, it's telling us how psychologists and linguists are spending their attention. What are they spending their time on? What are they paying attention to? That really doesn't tell us anything scientifically about why they're doing that, why it's important, what the knowledge gaps are that they're trying to address. It's very indirect. And so it's leaving the readers to assume, and you don't know what your readers know in terms of background information. So they might assume the wrong thing and they'll be completely confused. His fix for this is to revise this first sentence as such. All children acquire the ability to speak a language without explicit lessons. How do they accomplish this feat? Now, that revision is very clear and it's very direct. It's much easier to understand. Let's now look at some introduction logic templates. So these templates are going to cover probably almost any research paper. And so we're going to look at 4 different templates here. The first one is we'll write some paragraphs or sentences that include facts about nature. Next, we'll talk about how the existing work explains these facts poorly. And then we move into talking about how, in this paper, we look for a better explanation. A second template is the topic is important and extensively studied, but something is missing, and we fill in the gap. A third template could be something like this. Theories or facts seem contradictory. Our experiments will resolve the tension. And finally, society needs X technology. Current technology has this problem or limitation, and we improve the technology to overcome the problem. So what you do with these templates is basically write out the information for your specific situation that fills in the facts about nature, about the existing work, about why the topic is important, and so on. Now let's look at some bad introduction templates. And these are based on papers I have actually read that set up their introduction in ways that you see coming up here. First, the topic is important and extensively studied. Then they write a miniature review article on the topic, and then they did something related to the topic. What's wrong here is that we don't see what the knowledge gaps are, and so the research question is not well defined, and it doesn't seem important because we don't know why they're doing this research. And, I must admit, in the past, before I learned a lot about scientific writing, I wrote some papers that were kind of like that, and I had a hard time getting them published. And now I understand why. A second bad template would be something like this. The topic is important and extensively studied. A, B, and C are missing, but we're going to study D. So what's wrong with this template? Pointing out all of the missing knowledge in a field is not something you want to do in an introduction to your paper because it's confusing to the readers. They don't know which of those knowledge gaps you're going to be addressing later in the paper. And so it's very confusing. They don't know what to expect, and so their expectations are not going to be met, and they won't be satisfied readers. So you should only point out knowledge gap D if you're going to study knowledge gap D. The third bad template is, this topic has never been done before, so that's why we did it. Now, it is good to do something that's never been done before. That's not a problem. The problem is it's not a scientific reason. There's not a scientific premise behind doing something just because it's never been done before. Now, how do you state your research question or purpose precisely and completely? So make sure that you are naming the variables that you studied, the materials that are going to be used, and how you're going to take measurements. Your purpose should not be look and see. So we're going to treat the animals with this chemical and look and see how they respond. That's what we call a fishing expedition. It's not based on a scientific premise, so it's not very popular to reviewers for this kind of a setup. The question should be the logical conclusion of filling the gaps or the unknowns or solving the problem. And a lot of times I'll see introductions that are not written in this sequence of logic. So an example of a nicely stated research question is as follows. The main objective of this study was to assess the feasibility and accuracy of selecting low cadmium wheat lines during grain filling using cadmium concentration in the terminal tissues. So it's measurable, it's telling the materials that are going to be used and which stage and what is going to be measured. Now when you get to that paragraph where you give a preview of the experimental approach, you want to keep it brief. You're not going to be presenting materials and methods in this paragraph. You're just going to give an overview of the approach. So you can do that in 1 to 3 sentences. When you give this overview, tell what it is that you studied, the name of the organism or the name of the enzyme or whatever it happens to be, and describe how you examined it with a general technique. Don't go into a lot of specifics at this point. That's going to happen in materials and methods. So here's an example of how to do that. In this study, we took a genetic approach to examine iron uptake of naturally occurring mutants of the IRT1 gene in Arabidopsis. So it's giving the approach and what the approach is being used on, and so it's a general technique, but it's not a lot of materials and methods. So again, at the end of the introduction, you want to talk about how this work is going to be significant in your field and what are the implications of doing this work. So what you want to do is have an exciting closing sentence of the introduction. This is where you're going to put the significance or implication. This is going to keep the readers interested and encourage them to keep reading the rest of your paper so that they'll eventually cite your paper. And it also gives them additional context for interpreting the results and understanding the discussion. Here's an example of a significance sentence at the end of an introduction. The results of this study increase our fundamental understanding of cancer biology and will lead to predictive tools that will inform specific personalized treatments. So what are the common problems of an introduction? Now that we've talked about how to do it and a little bit about how not to do it, let's look at some of these common problems. First of all, a common problem is having missing elements or misstated elements or obscured elements. And we had an example of that earlier with the psychologists and linguists turning their attention to something. Another common problem is writing a mini review article that does not funnel to the research question. So that leaves the readers confused about where you're going with the article. Another common problem is that the introduction is too long and it's rambling and it's going off topic and it's talking about things that might lead the reader to expect that they'll be addressed later in the paper, but they're not going to be addressed later in the paper. So keep it short and keep it focused. The fourth common problem is that the context or background doesn't match well with the eventual objectives. Now we have reached our action list for this week. All right, so what do you need to do this week to write the introduction section? Let's take a look. First, and I want you to do this first, is to clearly define your research question or purpose. Now, that's not as easy as it sounds. And in practice, sometimes the research question that you're going to write your paper with is not the one that you started doing the experiments with. Sometimes it changes as you find the results, or it can change as you think about how to present these results. It's not being dishonest to write a different research question at this point than you started out with when you set up the project and began doing it. That's done all the time, and it's helpful to your field and readers to present it in a way that they can understand rather than a way that what you did doesn't match up well with the research question. Action list item number 2 is, as you write the introduction, set the context in the first paragraph. Item number 3, in the first few paragraphs, give the background and funnel down to the knowledge gaps. Then state the research question or purpose in place in the paragraphs of the introduction section. So you defined it earlier as you were doing your pre-writing for this section. Now you're going to state it, and you're going to break that research down into objectives. So how do you go about accomplishing answering this research question? You do that by carrying out some objectives. And then you're going to finish off the section by explaining the significance of your study. Let's wrap up this lesson by looking back at the learning outcomes. So by the time you complete this lesson, you will be able to explain the purpose of an introduction, explain the key elements of an introduction section, structure your introduction section to funnel from general to specific, signpost all the key elements of the introduction, and avoid the common problems of the introduction section. Make sure you have accomplished all of these learning outcomes before you move on to the next lesson. And happy writing! Welcome to the lesson on materials and methods. Let's get started by taking a look at where we are in our project management approach to writing this paper. So we are in the executing phase as we were writing the introduction. And as we are executing, remember, we're also controlling and monitoring. So we're looking back to our writing plan, the outline that we constructed earlier, and we're deciding if we're on track, and we're deciding if we need to make any changes. So these are the controlling and monitoring steps of the process. Let's look at the learning outcomes for today's lesson. At the conclusion of this lesson, you will be able to explain the purpose and content of a materials and methods section. You will be able to explain what information to include to make a complete description of materials and methods. You will be able to provide the appropriate level of detail, and you will be able to provide correct references as needed. Also, you'll be able to avoid the common problems that happen in the materials and methods section. Let's begin by taking a look at the functions of the materials and methods section. So the first function is to detail the materials that you used in the work. So a lot of you in this course are going to be in life sciences, and so the materials that you choose are very important. The study organisms are very important because different species obviously are different. Different strains or varieties are going to behave differently than other ones. And if you're in some other area of science, physical sciences or chemistry, for example, the materials are also very important in your case. So one of the main functions is to very specifically explain which materials you used in your study. You're also going to describe your experimental methods. And we'll get into some details later about how thoroughly you should describe these methods. But it's really important to do so in a way that's at least thorough enough that another scientist in your field could go and replicate the study. In other words, they could take your materials and read your paper and do the same experiments and come up with results that are very similar, if not the same. Also, the materials and methods section is going to give context to the results that are coming later. And so when the readers are looking at a figure and they want to understand how that figure was generated, they'll go back to the materials and methods and find the appropriate paragraph and read it, and then they'll understand that figure better. It's also going to assure other scientists that you did the work in a valid and rigorous way. That's very important. Reviewers are going to look very carefully at how rigorously you carried out your experiments and if you used valid materials and valid methods to get these results. And the final point, which I've already mentioned, is the function of the materials and methods section is to allow others enough information to reproduce the results. So what should you put in the materials and methods section? So for materials, these will be things like drugs, culture media, buffers, gases, apparatus, locations that you studied or specific sites. It could be patients or subjects, experimental materials, animals, microorganisms, or plants. In terms of methods, you need to include information on your experimental design. So mention your independent and dependent variables, your experimental and control groups, and any other pertinent information like randomization. And also talk about the procedures, what you did, how you did it, and sometimes you need to explain why you did it. Sometimes that is obvious, or it should be in the context of the paper, but sometimes it's not obvious and you need to add a little bit of an explanation for why you chose this particular method. Let's look at some tips for writing the materials and methods section. Now, in terms of writing style, this section is the only section in the paper where the passive voice is preferred. And so if you don't know what I mean by passive voice, I'll have you go and look that up. But it's basically the way that you choose the verbs. Secondly, in the materials and methods section, you want to organize this section using subheadings. So that's going to greatly help your reader follow what you're doing and help them quickly find information when they want to refer back to this materials and methods section. So break it up by subheadings. And also think about organization of this section based on the objectives that you presented in the introduction section. And those objectives came from your research question. And this is going to help you decide how to organize the paper and how to choose which subheadings you're going to list. So materials and methods are pretty straightforward, right? So you might have this question: can I just copy and paste my materials and methods from a previous paper that I wrote when I'm doing the same things as I did before? Well, I don't think you should do that. Now, as we mentioned when we talked about plagiarism, in materials and methods, a word-for-word sentence that appears in more than one publication is not necessarily plagiarism. However, I do suggest that you don't just copy and paste. I really suggest that you write a new materials and methods section every time you write a new paper. Then, in the chance that some of the sentences are the same, you don't need to worry too much about that. But I do recommend writing it freshly every time. Some backup for this approach is from the Committee on Publication Ethics, and they have this quote on their website that says, "Use of similar or identical phrases in methods sections where there are limited ways to describe a method is not unusual. In fact, text recycling may be unavoidable when using a technique that the author has described before, and it may actually be of value when a technique that is common to a number of papers is described. Editors should use their discretion and knowledge of the field when deciding how much text overlap is acceptable in the methods section. The reuse of a few sentences is clearly different to the verbatim reuse of several paragraphs of text." Now, you might be thinking to yourself, well, the materials and methods section is so descriptive, how can I possibly mess it up? What could go wrong? Well, there are a few things. You could provide too much detail or too little detail. Yes, it is possible to provide too much detail, and that's when it's going to become confusing to the readers because they don't know which things that you're saying are important and which things that you're saying are not important. Or it might just be obvious. You don't have to explain how to make a sucrose solution. People should know how to do that. Another way that could go wrong in the materials and methods section is not telling the purpose of an experiment if it's something that's unusual or not obvious. If it's something that you invented, no one has done it before, you might need to give a little bit of an explanation for why you're doing this method or procedure. You could also leave out information, and this is different from too little detail in that you just completely didn't tell about that part of what you did. So it's just completely missing a paragraph or a few sentences. Or you could give incorrect information. And I actually had this situation. A student of mine came to me one time and he said, "I'm trying to grow these plants and I'm using this method from this previous publication. And the plants don't look healthy at all. There's something wrong with them. And so I said, okay, let me take a look at the paper that you're getting this method from. And I started reading it, and I read the nutrient solution composition. And in one of the micronutrients, they had a typo. And this typo resulted in my student using 100 times more of a certain mineral than they should be using. And so, of course, 100 times more is in the toxic range. And that's why his plants were not growing, because he was following this recipe from the publication, but it was incorrect due to a typo. Let's look a little bit more at materials. What should you do with materials? Well, make sure you define them very precisely. In other words, if you're using a certain chemical that could have different levels of hydration, make sure you're listing the correct one. If you're using a certain strain of a mouse, make sure you list the name of that strain. It's also a good idea sometimes to tell where you got the materials. So you will include trade names or manufacturer information. But you only need to do this when the product is unique. It's only available from that specific manufacturer, or if getting it from that manufacturer distinguishes it from a different manufacturer. Use systematic names for chemical substances as needed. So there might be an abbreviation for a chemical substance, but on the first use, go ahead and spell out the exact long chemical name of that substance, just so there's no ambiguity. Also include information such as the total number of subjects or samples, the number of subjects receiving treatment, how the subjects or samples were selected, and any details about them at the time of treatment or data collection, such as the sex, the age, or whatever is important about those samples. Now, when we get into methods, a lot of times you'll talk about the equipment that you used. And just like with materials, you only need to include the manufacturer information if that equipment is different than the equipment produced by a different manufacturer. So if you're using a centrifuge, you don't need to specify whether it was made by Eppendorf or made by Beckman. Those two brands centrifuge things exactly the same way if you're using the same G-force. So you don't really need to go into that amount of detail. Do give precise technical specifications, such as what temperature you were using, the pH, the total volume, the time, the quantities, anything else that is important in the procedure. Go ahead and specify that, and put the methods and sequence of procedures in an order that makes sense to the readers, that they can follow clearly. It doesn't necessarily have to be that every section is in chronological order. In fact, making everything in chronological order could be very confusing. Also, don't forget to describe controls and control experiments. Positive controls and negative controls are so important, and so you need to include that information also. Make sure that it's complete. Now, one thing I've noticed with students who are writing for the first time is sometimes they tend to write the methods section as a set of instructions. And that's not what you're doing. You're writing in the past tense and you're explaining what you did. You're not giving instructions to someone to do something later. Okay, how about references in the materials and methods section? You will have a lot of references in this section. For previously described methods, you only have to give the reference, but if you want to be nice to your readers and considerate to your readers, it's good to give some additional information. For example, I could say ferric chelate reductase activity was determined for whole intact root systems as described previously, Romera et al. 1996. So that meets the requirement, but it's really not enough information for my readers to fully understand what's going on. If they want to understand it more completely, they would have to go to this paper, look it up, and read it and find out what was done. And sometimes that turns into a chain where that Romera paper may have only cited a reference to another earlier paper, and that paper may have cited only another reference to an earlier paper. And so you might have to go back 4 or 5 papers before you find a full description. So I believe you shouldn't do that to your readers. Go beyond the basic requirement and do something more like this. Ferric chelate reductase activity was determined for whole intact root systems as described previously, Romero et al. In brief, roots were rinsed and submerged in 70 ml of assay solution. Here's the recipe. After 20 minutes, an aliquot of the assay solution was removed and absorbance at 535 nanometers was determined. Iron 2 BPDS concentration was calculated using the extinction coefficient. So now you can see we've added a little bit of information that tells the approach of what we did while also citing the original reference. Now, certain techniques or procedures have guidelines that have been published. For example, there is the MIQE. This is the minimum information for publication of quantitative real-time PCR experiments. So you can go onto Google Scholar or Web of Science and look up MIQE and get this publication and follow those guidelines when you write your materials and methods section. Similarly, there is Miami, minimum information about a microarray experiment. There is MIGS, minimum information about a genome sequence. There's also minimum information about a marker gene sequence and any sequence. And so all of these minimum information guidelines will be really helpful to you to know what to put in your materials and methods. Additionally, if you're doing human subjects research or you're doing animal research, you have to have approvals by your institutional review boards or other review boards to do this work. And so you need to give that information, such as the permit number, in the materials and methods section also. Okay, we have arrived at the action list. And so this is your action list for the week for materials and methods. The first thing is you need to gather descriptions of all your research sites, gather all your protocols, and gather descriptions of your key materials, your genetic stocks, your cell lines, your chemicals, your seed stocks, whatever it is that you have. Fourth, make a list of any proprietary materials, including the manufacturer and their location. Make a list of all special equipment with the manufacturer and location. Describe the conditions for procedures that have specific conditions. This will be most of the procedures that you carried out. And then figure out how to organize the section to tell what you did and how you did it by using subheadings. So choose the subheadings that you're going to use in this materials and methods section. And then go ahead and write the materials and methods section. Write up these paragraphs. That was your guidance for writing the materials and methods section. Before we finish, let's briefly review the learning objectives for this lesson. At the end of this lesson, again, you should be able to explain the purpose and content of a materials and methods section. You should be able to explain what information to include, provide the appropriate level of detail and the correct references, and avoid the common problems of the materials and methods section. All right, you have a lot of work to do this week, so go ahead and get started on materials and methods. And happy writing. Hello and welcome to the lesson on the results. Let's look at where we are in the project management approach by looking at our project management diagram. So here we are, we're still in the executing phase. We are also doing controlling and monitoring, looking back at our writing plan. It's okay to make changes to your writing plan as you go. Just make sure you're making those changes intentionally and not just getting off track because you're not paying attention. The learning outcomes for today's lesson are as follows. You should be able to explain the results of your study without repeating the materials and methods. You should be able to explain the results of your study without presenting discussion material. You should be able to explain the results of your study without repeating the underlying data. And so we're going to get into the difference between data and results shortly. Also, you will be able to refer to figures and tables rather than just announcing them. You will also be able to write appropriate figure legends for a results section and to avoid the common problems in the results section. So what are those common problems in the results section? Well, the first one is that there are missing components or missing signposts, or missing transition phrases. And so this is going to make it hard for the readers to follow where you're going with the results section. You could also have excessive experimental detail in the results section. In other words, you are repeating the materials and methods as a way to lead up to what the result is going to be. And we'll talk about how to avoid doing that while still giving the reader some context for the results. Another common problem is including comparisons, speculations, and conclusions. So comparisons to other published research, for example. All of these things should be in the discussion section. They should not be in the results section. So that's why it's a common problem. And we need to keep those two sections separated from each other. Unless you're writing a combined results and discussion section, then this does not apply. Next, I would like to give you some tips for writing the results section. You want to use past tense for your results that are specific to your study. There may be times when you're setting the context that you refer to results of another study to explain why you did a certain thing that gave you this result, in which case you might use a different tense. However, when you're referring to your own results, these are things that were done in the past. And so you want to refer to them in the past tense. An example of this is you would say iron deficient Arabidopsis plants had chlorotic leaves, because you're describing a specific situation that you observe, and you don't want to be generalizing to all Arabidopsis plants at this point. Now you also might use the present tense for certain results that are always going to be true. For example, the karyotyping results showed that Arabidopsis has 5 pairs of chromosomes. So your results showed that, but you expect that that's always going to be true. So you can state that in the present tense. So think very carefully about tenses in the results section. Most of the time you need to use the past tense. Rarely will you use present tense. Another tip is to use subheadings. You used subheadings in the materials and methods section. I would suggest using them again in the results section. These are really great organizational tools. At least use them in your drafts. Now, you might want to remove them when you go to submit your paper, because some journals don't permit subheadings in the results section. Also, remember to use transition phrases. These are words like however, because, therefore, in addition. They're signals to the readers. They're going to guide the readers when you're changing topics, when you're making a comparison or a contrast statement or a contradiction. However, you don't need to go back and repeat introduction material as a transition, and you don't need to bring in discussion material as a transition. What do you put in the results? Are you going to take your entire lab notebook and put everything that you did into the results section? No, you're not going to do that in most cases. You're probably going to put only the most important findings and results. You're not going to include every result that you generated during the project. For example, you probably did some pilot studies to optimize your treatments, or the timings of your treatments, or to optimize your measurement techniques. And so including all of these things is probably not going to be helpful, and it might even be distracting. Now, I'm not suggesting that you cherry pick your results and only show results that lead readers down a certain path and tell a certain story that you have predetermined. That's not what I'm suggesting at all. I'm just saying that some of the work that you did is just not going to result in publishable results. Now, you might have some results that are not entirely necessary for your scientific story, but they are helpful, and you can put those results into supplementary materials. So let's look at a little bit more detail at signaling and signposting. One thing that's important to do as you write is to signal to the reader which results are the most important ones. These are the ones that provide new knowledge. But at the same time, you can't just leave out results. So you've got to figure out ways to signal and signpost which results are the most important. What you're doing in the results section is you're interpreting the data that you collected. Data are not equal to results. Data are the raw data points that you collected, and you analyze those by fitting a curve, or by using some software algorithms, or by calculating means and standard deviations. And so you've converted those data into results. Interpreted data equals results. So you have put some meaning onto the data. Now, we don't want to go into the discussion section and the results section. What's the difference between interpreted data and discussion? Well, interpreted data is looking at it in the context of the objective and the procedure that's being done. When you interpret those results to the context of other research that's already been done, or into the context of the research question or application, that is interpreting results to make a discussion point. So this might be new information for some of you. When I've told my students that data and results are not the same thing, they were pretty surprised by that, and they'd never thought about it before. So don't use these terms interchangeably. They don't mean the same thing. So one thing that is typically difficult for students is describing their results. And they have a lot of trouble describing their results without repeating the data. So how can you describe your results? What kinds of things should you be looking for? One kind of result that you can pull out of your data could be a general classification, such as the phenotypes may be divided into 3 main categories: dark green, pale green, and chlorotic. You can also make comparisons and contrasts. Mice in X treatment were twice as large as mice in Y treatment. You can also describe trends. After treatment began, there was a sharp decrease in survival, followed by a plateau. Also, you can describe high or low points. Growth rate peaked at 21 days after pollination. So these are all kinds of different ways you can pull results out of your data. Some additional ones are describing ratios and proportions. Plants with high nitrogen treatment had 125% higher yield, or phenotypes for flower colors segregated in a 3-to-1 ratio. You can describe fractions, such as 30% of the plants survived the drought treatment after 3 weeks. You can describe averages. The mean grain iron concentration was 35.6 micrograms per gram across 299 lines. You can describe ranges. The grain iron concentration ranged from 19.5 to 67.9 micrograms per gram. You can also describe causality. The plants with a mutation in the FIFI gene failed to induce FRO1 gene expression, or higher nitrogen supply led to increased growth. So those are all type of descriptions that you can try to make. And there's probably other ways. All right, here's a question for you. Fill in the blank. Simply restating data from a figure or table is not presenting a result because of why? What do you think? If we look at this figure and we have this sentence in the results section, grain yield was 2.6 grams per plant for wild type and 3.6 grams per plant for the RNAi line at 18 milligrams nitrogen supply in the optimal environment, and about 0.6 grams per plant for both lines in the heat stress environment. So this sentence is simply repeating the results from the figure. They're not giving us any information that we can't see ourselves if we just look at the figure. It's repeating, right? It's not interpreting. So how would I fix this? So I would revise this sentence by saying grain yield per plant was significantly higher for the RNAi line at 18 milligrams nitrogen supply in the optimal environment. However, yield for both lines was decreased by 8 to 10-fold in the heat stress environment. So I've done two things there. I've done a comparison between the two lines, and I've also looked at a proportion or a fold change between the treatments. So I've interpreted this data in the context of the experiment. Now, another skill that you should develop in the results section is referring to the results in the figures and tables, not just announcing that they are there. Do this by referring to the figures and tables in parentheses. Let's look at an example where this is not done very effectively. The concentration of zinc in group C and in the control group is shown in figure 1. Okay, so this example is just simply announcing this is what's in figure 1. It's not really telling us anything useful. How could we do it better? We could say something like this. Zinc concentration was lower in group C than in the control group, figure 1. So what I've done here is I've given a result and then told the readers where they could see the figure that contains that result. So do you see the difference between announcing and incorporating the reference to the figure in a result? This slide is adapted from a post I saw on Twitter from the Dynamic Ecology blog. It says, quick quiz. Let's imagine you are reading the results section of a manuscript. Which of these is the most useful / interesting / compelling / informative? Choice number 1. Figure 2 shows the relationship between infection and lifespan. Choice number 2. Our experiment on the relationship between infection and lifespan found unambiguous results. Figure 2. Choice 3. Including infection treatment as a predictor improved model fit for lifespan. Statistics and figure 2. Choice number 4. Infected hosts lived, on average, half as long as uninfected hosts. 20 days versus 40 days. Statistics, figure 2. So I think most people are going to choose number 4. Choice number 4 is the most skillfully done result sentence. It's interpreting the results in the context of the experiment and referring to the figure in the same sentence. Whereas choice number 1 is simply announcing it. Choice number 2 is giving some information that's not useful whatsoever. And choice number 3 is just making a statement about the procedure. Now, we mentioned statistical support in that last slide. Make sure that you do not present the statistical support as if it were the result itself. So the statistic, like a p-value, for example, is not the result. It is supporting the result. Now, I mentioned earlier when we were looking at learning objectives that you need to not repeat materials and methods in the results section. When authors do that, usually the reason is that they're trying to give some context for the result they're about to tell. Instead of repeating materials and methods, what I recommend to do is use this repeating pattern. Briefly tell the purpose or background of the experiment, and then give an overview of the experimental approach, but not materials and methods details. And then give a results and an interpretation if necessary, which is the answer to the objective, but do that without going into discussion. So again, the interpretation needs to be explaining the result in the context of the experiment, but not explaining the result in the context of the research question. Now the figures are going to appear in the results section, so the figure legends are part of the results section. One thing to keep in mind is that figures and tables should be able to stand on their own. In other words, a reader should be able to just open the paper and look at a figure and its legend and be able to understand what you did, how you did it, and what it means. So you need to have a lot of information in the figure legend to accomplish this important task. So the parts of the figure legends that you need to have are a brief title, a description of the figure content, the approaches you took to generate the data in those results, definitions of symbols and line or bar patterns, abbreviations that were not defined earlier in the legend, and for graphs, statistical information. For example, what do the error bars represent? Are they standard deviation? Are they standard error? What do the asterisks represent? Are they a certain p-value threshold? Let's take a look at an example. So we have this figure in a results section, and we have this figure legend that says, figure 1, cucumber roots do not respond to iron supply in alkaline nutrient solution even though they are chlorotic. Panel A, plants in each treatment. Panel B, chlorophyll level. Panel C, ferric chelate reductase. The question is this figure legend adequate? Does it have all the information that I just told you needs to be in a figure legend? Clearly the answer is no. This figure legend is not adequate. It does not contain all of the information. What was the actual figure legend for this figure? And you see it here. Now, I'm not going to read this entire thing to you, so why don't you go ahead and pause the video and read this figure legend and think about, does it contain all of that information that I told you should be contained in a figure legend? A little bit more about figure legends. First of all, as I just mentioned, remember that figure legends are in the results section. So write them as you would write results. Point number 2, with that in mind, begin with a caption that is a statement or sentence that describes the contents of the figure. Do not caption it with a conclusion about what the results mean. Why not? Because that is discussion material. Interpreting what the results mean in the context of the research question belongs in the discussion. The figure legend is in the results. So don't lead the reader to making a certain conclusion. Give them the opportunity to study the figure or table and come to their own conclusion. Then when you get to the discussion section, you can tell them your conclusion about those results. But don't do it here. Let's look at some examples. Now it's time for the action list for this week. First, I want you to revise and finish your figures and tables. So the figures and tables are going to be the heart of the results section. So now is the time to go ahead and finish those. Next, I want you to write the figure captions. So a one-sentence description of what's in the figure. And then next, I want you to go ahead and write the complete figure legend. So look back to the slide where I showed you all of the parts that need to be in a figure legend and make sure you include all of that information. Next, I would like you to print each figure on one page, one piece of paper, and measure it with a ruler. And make sure that the width of your figure matches with the column width in your target journal, and then resize it as necessary. Now it's important to do this at this stage before you submit the paper, because a lot of journals will publish your figure at the exact size at which you submitted it. So if it's a little bit wider than a column width, they'll publish it taking up 2 columns, and it just doesn't look right on the paper. And also, some journals will shrink it to fit the column width, and if they shrink your figure, it might not read very well at that point. So this is a good time to print it and make sure that you can still see all the symbols, that they're large enough, that all of the letters and fonts are large enough when it's printed at column width. Next, I want you to draw on your figures. You're going to draw trend lines, circle important data points, and just kind of figure out what it is that you are seeing that allows you to interpret that result. So what is it that you want to help your readers see in that figure? What is the important thing to describe? So just mark that up on the figure itself, and that's going to help you when you get to typing up the results. Next, on the printed figures, write down the key points you need to make. So you might have one key point from the figure, or you might have 4 or 5 key points that need to come out of that figure. Go ahead and jot these down with pencil and paper onto the figures that you have printed out. Next, decide the order that you're going to present the figures. And now you're ready to write a results paragraph for each figure using the repeating pattern. And once you have all of these results paragraphs written, you've got a full first draft of the results section. Okay, there was a lot of information there in that lecture. Results seem like they're a straightforward section, but they are pretty tricky to do well. So let's look back at our learning outcomes and make sure that you can do each of these learning outcomes before you move on to the next lesson. Okay, learning outcome 1 is to explain the results of your study without repeating materials and methods. Number 2 is explain the results of your study without presenting discussion material, including in the figure legends. Number 3 is explain the results of your study without repeating the underlying data. So remember, data do not equal results. The next one is refer to the figures and tables rather than announcing them. So skillfully incorporate that reference to the figure in results sentences, not in announcement sentences. Write appropriate figure legends for a results section And finally, avoid the common problems of the results section. All right, so this is another week where you have a lot of work to do, but I have faith that you can do it. So get out there and write this results section. And as you do it, happy writing. Welcome to the next lesson. Today's lesson is about writing the discussion section. So we're getting close to the end of the main body of the manuscript, and today we're going to talk about what should go into the discussion and how to write it and the common mistakes that you should avoid. So let's get started. First, we're going to look at where we are in the project management process. So we are here still in the executing phase, and as you are executing, writing the discussion, you should also be controlling and monitoring. That is, looking back to your outline, making any adjustments that you feel like you need to make as you're going, but not drifting off course aimlessly, making these adjustments purposefully and adjusting your course as you go. The learning outcomes for the lesson today are as follows. At the conclusion of this lesson, you will be able to explain the purpose of a discussion section. You will know how to structure the discussion in a pyramid form, going from more specific information to more general information as you move through the section. You will be able to discuss the findings of your study without repeating the results material. And this is a challenge for a lot of new writers. You will also be able to discuss your findings and prior related publications as opposed to just citing them. You're going to discuss them in the context of your research question. You will also be able to refer to the figures and tables as appropriate, and you'll be able to discuss the significance of your study and propose future directions. And you'll be able to avoid the common problems of the discussion section. So I'd like to share with you one more quote from Mr. William Zinsser, and he's talking about how to end an article. So before we had a quote from this same author talking about how to begin an article. Now he's going to tell us a little bit about how to end an article. And he says, knowing when to end an article is far more important than most writers realize. You should give as much thought to choosing your last sentence as you did to your first. Well, almost as much. So let's translate this quote into scientific writing. I told you that you should put a lot of thought and effort into the introduction section, which is true. I still believe that. But you should also put about as much thought into the discussion section. So this is a really important section for your readers because this is where you're going to synthesize your findings from results into knowledge. So you're going to go from data to results to knowledge, and that happens in the discussion section. And so it's really important that you have a good plan, a good outline, a good structure, and your logic and your reasoning flows throughout this section. Also, that last paragraph of the discussion can be a really powerful paragraph. So make sure that you write a conclusions paragraph that's going to end the article with some excitement. So the readers are agreeing with you. They're saying, yes, that is the future direction that this research needs to go. All right, so what are the functions of the discussion section? One of the first functions of the discussion section is to provide an answer to the research question. That's really important. The research question has been the central theme of your entire manuscript, and all the work you've done is to reach an answer to that. And so this is where you're going to provide the readers with that answer. It's also going to explain how the results support the answer to the research question. And it's going to place the answer to the research question in the context of existing knowledge. At the same time, it's going to create new knowledge. And it's also going to provide the theoretical implications or practical applications of your work to future situations and how other scientists in your field can use your findings to advance their work also. Now let's get into a little bit of the writing details. As you write the discussion section, you're going to be talking about, in some cases, the results that you found. In some cases, you'll be talking about results that other scientists found previously that are already published. And in some cases, you'll be talking about future directions. So you're going to have different verb tenses in this section. So one of the common mistakes that people make in the discussion section and in the results section is to discuss the results of the current study in the present tense. When you're talking about your own results that you generated during this study, most of the time you're going to be talking about them in the past tense. The only time you would talk about them in the present tense is similar to what we talked about in the results lesson, and that would be when your discovery can be applied to all situations. And that's not very common. Most of the time you're going to be discussing your results in the past tense because this is what you did find in the past before you wrote this paper. Now when you talk about other people's work, since it has been published, we consider that part of the knowledge base, so that should be discussed in the present tense. When you get into the final paragraph, or any time in the section when you're talking about future work, then you can use the future tense for the verbs in those sentences. So how long is your discussion section going to be? Now, at the beginning of the course, I gave you an assignment where I wanted you to go out and survey the literature in your field and get some baseline information about how long these sections typically are. So think back to what you found there. What's normal for your field, or what's the range that is expected? Also, keep in mind that the discussion should be no more than 1/3 of the body of the manuscript. It is going to be the longest section a lot of times, but sometimes authors tend to make it way too long. And they're just over discussing things, and they're going off on side directions that are kind of relevant, but they're not directly relevant to this study, and it just gets out of hand. So a lot of times when you get reviews back, the paper's too long, and they want you to cut it. I've had reviews come back where they ask me to cut the paper by as much as 1/3. A lot of the cuts will come out of the discussion section. So try to keep it to 1/3 at the max. And for most papers that I've seen in the life sciences, 5 to 7 paragraphs is adequate to accomplish everything you need to in the discussion section. So as you're putting together the discussion, what are the components that you're going to have there? Let's take a look at that. One of the key components or the key elements is going to be the main take-home message, which is the answer to the research question. And that's going to be in the first paragraph of the discussion. You're also going to have comparison and contrast to previous studies. Quite a bit of the discussion will revolve around that. That's how you're going to figure out what your results mean, is by comparing it to what is already known in your field and what previous authors have found in studies that are related to what you're doing. You should also have a paragraph that goes over the strengths and the weaknesses of your study. You can talk about unique approaches that you might have done, new approaches that maybe no one else has ever done before. So basically a critical assessment of your study. And if you know your study has some weaknesses, you should point that out. Don't wait for the reviewers to point that out, because if the reviewers point it out, it's more likely to push you towards the reject decision. But if you can get in front of it and say, we recognize that our study has these weaknesses, but we had to design it in this way because of XYZ, then the reviewers might be a bit more forgiving and understanding that there are these weaknesses. So you just can't make a perfect study. And other scientists recognize that. So having this critical assessment is going to help you in reviews by taking away some of the points that reviewers might criticize otherwise. You're also going to have a section with conclusions, significance, and future directions. And we'll get into some more details about that last paragraph later in this lesson. Other features of the discussion, in addition to those key elements we just discussed, are that you should discuss other relevant results and hypotheses. So not just the main answer to the research question, but you have other results in the study also. Go ahead and discuss those. Mention any possible errors. This is going to complement discussing the weaknesses of your study design, but it's a little bit different because this is focusing on the technical aspects. Maybe you had to use some instrumentation that has a certain error rate in it, and you might need to account for that as you talk about what your results mean. Point number 3 is you should also discuss any unexpected findings. Unexpected things happen all the time in science. And, sometimes you might feel like it's taking you off of your plan that you had so carefully designed. But these unexpected findings can be really exciting. Think about if the unexpected findings are bad for your study, then you can go back and rethink the research question so that the unexpected findings actually turn out to be good for your study. So don't try to minimize or hide any unexpected findings. Bring those out to the forefront and go ahead and discuss them. Also, indicate any new questions that arise. Now, this might be part of your future directions, but there might be other new questions that pop up. So now that you have discovered this specific thing that you discovered, you now have filled that knowledge gap, but maybe a new knowledge gap appears. And so this is something that readers get excited about when you have discovered a new knowledge gap as part of your analysis. So go ahead and point those out as well. Now, the whole point of publishing papers is to have impact in your field. So what does impact mean? Well, part of what impact means is citations. So you want other people to cite your work. That's one way to measure impact. Another way to measure impact is if people change their behavior after your work is published. So maybe your work is informing a policy or a common practice in your field, and people start doing things differently because of what you discovered. In that case, that's a very high impact. How do you show the readers the potential impact of your work? So there's a few things that you can do in the discussion section. Your work is going to be impactful if you can challenge an accepted idea, concept, or current paradigm. You will set that up in the introduction, but the discussion is when that comes to fruition. And if your results are showing that this current thought is not quite true, and you've contradicted it and you found the real truth, that's very impactful. Secondly, answering an unsolved question is very impactful. Now, of course, you're going to answer some unsolved questions. That's the whole purpose of setting up your research question. But maybe your research question is focused on something that has been a long-standing mystery in your specific field. So it's a big question that a lot of people have. So that's going to be more impactful than it's a very specific, small question that only a few people in the field have. This third one is related to that last point that we just talked about. If you can present a new finding or discovery that fills an important gap. So this is more geared towards the technology-driven type projects, where there's maybe a technology gap that you've discovered a new method or a new instrument or a new way of doing things that fills that gap. And finally, if during your discussion you can come up with the need for a new model, or even better, if you develop a model to explain your findings in the broader context. Having that model figure usually falls in the discussion section rather than the results section, and it's really powerful. Readers really like to see a model explaining the findings of their particular study. And usually that model will lead to a lot of new questions, a lot of new research questions, or a lot of new knowledge gaps will come out of such a model. So that will really make your work impactful if you can do that. I wanted to just put up a slide to remind you of the overall structure of a scientific paper in the graphical representation, which is the hourglass figure that we talked about before. We've already gone through the introduction and the materials and methods and the results on this hourglass figure. So now we're getting into the discussion section. And you can see it's kind of a mirror of the introduction section. So whereas the introduction section started with general information and became more specific, the discussion section is going to start with specific information and become more general. And the way you're going to set it up in this pyramid structure is to have the major new knowledge right at the beginning. And as you discuss that major new knowledge, you're going to reflect back to the research question, reflect back to the objectives, and discuss the answer in the context of those two portions of the paper that you had set up during the introduction. As you move through the discussion section, after the first paragraph, the next few paragraphs are going to be talking about the other new knowledge that you have generated. So other new knowledge 1, other new knowledge 2, etc. Also, as you develop through the discussion section, you're going to talk about how your new knowledge relates to the knowns. So reflect back to the knowledge gaps, and you're going to talk about how your results and findings filled the knowledge gaps that you pointed out during the introduction section. And finally, you'll end up talking about how your work creates new understanding. So you have to wrap all of this stuff into the discussion section. So it can be pretty challenging. And so this is why we're talking about all of these things in detail, so that you can be aware of what you need to accomplish in the discussion section. Let's elaborate a little bit on how to structure the discussion section. So as I mentioned, the first paragraph is going to contain the main take-home message, which is the answer to the research question. Now keep in mind that the answer is an interpretation of your specific results to generalize the findings to the broader context of your research question. You're not going to answer the research question by just repeating the results, but you do need to mention the results to support your answer to the research question. So again, discussion is interpreted results. As you answer your research question, you're interpreting your results, and you might be combining results from several different figures. You might have results from figure 3 and figure 5 and table 2 that when you put them together form the answer to the research question. It may not be one specific result, but a combination of those findings. So again, in this paragraph where you're answering the research question, it's coming from the results, but you don't repeat the results section. You need to interpret the results into new knowledge that answers the research question. And as you do that, you should refer to the figures that provided that result that you're interpreting. So refer back to figure 3 and figure 5 and table 2. So there's some common mistakes that I see frequently in the discussion section, and they are some ways that you should not begin your discussion section. The first of these is by writing a second introduction. I see that sometimes, and I've actually made that mistake myself in the past before I learned a lot about scientific writing and what to do and what not to do. If you need to bring in more material that you didn't introduce before, sometimes that happens, but you need to work it into the discussion at the appropriate place. You don't need a whole second introduction paragraph. Now, if you find that you have that second introduction, maybe you should move it into the real introduction. Or maybe it's just not relevant and you're just trying to find a way to get started. So a better way to get started would be by revisiting the research question and why you're asking it. And that's going to build up the momentum to answer the research question. But don't start with a second introduction. Also, don't start by summarizing the results. Okay, the readers just finished reading the results section. They don't need to read a summary of what they just read. Avoid that mistake too. Continuing on the organization of the discussion section, in the last paragraph, this is where you're going to have the conclusions, significance, and future directions. Now, as always, signpost the important elements or the important points that you're making. And for the conclusions paragraph, just start with the words "in conclusion." Make it easy for the readers to see that you're now wrapping it up and you're going to talk about some conclusions. It might be helpful in this paragraph to again restate the research question, but don't state it word for word. Paraphrase it and make it sound a little bit differently. And reiterate the significance of your work in the bigger picture in this paragraph. But you're not repeating what you've said previously. This should be a forward-looking paragraph, right? So you're looking towards the future. You're telling the readers what all this means in terms of going forward. So part of that is going to be saying what we need to do next. Now that you've done this work and you found some new knowledge gaps, next you need to address those knowledge gaps. That's an important thing to talk about in this conclusions paragraph. And end with a forward-looking final sentence. So how do you not in the discussion section? Well, don't write a summary. So a conclusions paragraph and a summary paragraph are quite different. The conclusions paragraph is looking forward. It's interpreting, it's building knowledge. And a summary paragraph is just repeating what you've already said. And it's not necessary to summarize. Don't end your section with a statement that just says further research is needed. That's not very interesting and it's not very informative. Of course, further research is needed. The research process is never going to end. We're always going to be discovering new things. Tell what further research is needed and why that further research is needed. And word it in a way that's more exciting than just saying further research is needed. Now, some of you are probably thinking, what if I need to write a combined results and discussion section? How do I do that? Well, we're not going to go into a lot of details about how to do that because the techniques are quite similar. And a combined results and discussion section is the norm in some journals. And it's optional in other journals, and some journals don't like it and they don't allow the combined results and discussion section. But for certain types of studies, the combined results and discussion section can be easier to write and it's easier for readers. So, for example, when I've written papers on functional genomics, like RNA transcript profiling, I found those papers easier to write and easier for readers if I do a combined results and discussion section rather than just a separate results and a separate discussion. The drawback with doing this is it's harder to incorporate all of the key components that you need in a discussion because the discussion is really focused on those specific exact results. And it's harder to work in the comparison and the contrast, and it's harder to find a good place to put the critical assessment of your study. You can do it, but you have to work a little bit harder to figure out how to do that. So if you do need to write a combined results and discussion section, what I suggest you do is to write the results section and then come back in and add the discussion to that results section. That has worked for me in the past. Now you could try to write them simultaneously. That might work for you. So if you do need to write a combined R&D, figure out what works for you. Now that you know how to structure your discussion section, you can avoid some of the typical problems that I see in discussion sections. A big one that I see is that the answer to the research question is not in the first paragraph. It might be buried halfway through the paper, it might be in the final paragraph, or maybe they don't even answer it directly and they leave it up to the readers to puzzle everything together. So that's a big one that I see. Make sure you don't make that mistake. Also, a mistake I see is that the authors do not point out the importance or significance of the study. So that's going to have less impact in the minds of the readers. Another big one is that the authors do a lot of speculating, and what I mean by this is they're including a lot of thoughts about how things might work, and they're making connections to very distant studies that are kind of related, but they're not directly related to the research question of your particular study. And so they're trying to do too much. This is kind of like in the introduction where a person writes a literature review instead of a background that funnels. This is the opposite of that, where instead of discussing the results in a very focused manner, they let the discussion get too broad and they're expanding it to too many tangential items. And so this is an inclusion of irrelevant information. Now it's relevant in the big picture, but it's not relevant to your study. What you can do to recognize this is look at your paragraphs. If none of your paragraphs are directly discussing any of your results, then that paragraph should be taken out. Another common mistake I see frequently is that the discussion is not really a discussion. It's just rehashing the results. It's repeating the results section, maybe with different wording, but it's basically just going over the results again. So make sure you're not just repeating the results, but you are doing comparisons and contrasts with previous studies and in a specific way, not just saying our results are similar to the results of this previous study, but explain why they're similar, how they're similar, and what that means, why it matters in the context of your study. And so that's what I mean by that final bullet point, that previous work in the field is not truly discussed, it's only mentioned and cited. All right, it's time for your action list. This is a big one. This is a big list. The discussion section is a really tough one to write. But if you did a good job on the introduction, it's going to make it a lot easier because the introduction is going to give you a roadmap for how to write the discussion section. All right, so let's go over the list. First, you're going to write paragraph 1. You're going to write the answer to the research question and support that answer with specific results and cite the figures and tables that those results are coming from. Then you're going to write the middle paragraphs, and you will write about the other important points to be made about your results. So you should discuss all of your figures and tables. And for each key point that you're making about all of your results, show how it relates to what is known. In other words, the previously published studies. Compare and contrast. And discuss how each result relates to the knowledge gaps or to the research question. Also, you're going to work in a discussion of the strengths and limitations of your study. That might be a standalone paragraph, or you might work that into various parts of the discussion, but be sure you have that included. Discuss any unexpected findings and why they're important. Make sure you're comparing and contrasting with previous studies. And if your study finds a totally different or a substantially different outcome than a previous study, it's important to bring that up to the forefront and discuss how yours is different and maybe why yours is different. Maybe you did the procedures slightly differently than that previous study, and that had a big impact on the result. Or, maybe the previous study did something incorrectly, and you can discuss that. You don't want to attack the previous authors, but you do want to do a detailed analysis of why you might have these contradictory results. So you're not going to discuss that in the results section, it's going to come in the discussion section. If your paper allows you to develop a model to explain your findings, this is where that figure is going to happen, in the discussion section. So go ahead and generate the figure and generate the paragraph discussing that model and explaining how it leads to new knowledge and new questions. Then in the final paragraph, you're going to write a conclusions paragraph. And also in that final paragraph, you're going to write about the significance of your findings, how they can be applied in your field, and then talk about what needs to be done next, the future directions. As we wrap up this lesson, let's take a look back at the learning outcomes. At the conclusion of this lesson, you will be able to explain the purpose of a discussion section. You'll be able to structure the discussion in a pyramid shape, going from specific information to general information. You will be able to discuss the findings of your study without repeating the results, and you will discuss your findings and prior related publications. do the comparison and contrast with those previous publications. You will refer to the figures and tables as you bring them up in the discussion. You'll be able to discuss the significance of your study, and you'll be able to propose future directions. And finally, you will be able to avoid the common problems of the discussion section. That concludes this lesson on the discussion. You have a lot of work to do, so get started on that. Enjoy the process, and happy writing. Welcome to the lesson on writing the title and abstract. Let's start by taking a look at where we are on the project management diagram. So we are still in the executing phase. This will be the last part of what we do in this executing phase, and then we're going to move on to the closing phase. We'll have a lesson on editing and revising and submitting, and that will be the final lesson. So we're at the next to last lesson. So let's get started. The learning outcomes for this lesson are as follows. At the conclusion of this lesson, you will be able to explain the purpose of a title. You will be able to describe and identify the types of titles. You will be able to write a complete, concise, and specific title. Explain the purpose of an abstract. You'll be able to describe all the key elements of the abstract. You'll be able to write an abstract that contains all of these elements and that conforms to journal requirements. And you'll be able to avoid the common problems of the title and of the abstract. Let's start with titles. What are the functions of the title? One of the main functions is that it provides the basis for readers finding your article. And so these days, they're going to find your article by searching a database like Web of Science or PubMed or Google Scholar or Scopus, and they're going to search it with certain terms. And if your paper has those terms in the title, it's more likely that the readers are going to have your paper come up as a top hit. So they're going to find your article, they're going to see the title first, they're going to read that title, and hopefully they'll become interested. They'll say, oh, that's an interesting title, I want to know more. And so they will click on the title, they'll see the abstract, and they'll read the abstract. So it's kind of a marketing tool. It's a way for readers to find you and get interested in what you're talking about. It's also going to let the reader know what the main topic and subject of your study is going to be. You might work on, let's say you work on Alzheimer's disease. Just working on Alzheimer's disease doesn't mean that every other researcher working on Alzheimer's disease is going to be interested in your paper. For example, if you're doing research on a diagnostic, researchers that are involved in treatment may not be interested in your diagnostic paper. Likewise, you may not be interested in their treatment papers. So having a title that gives the readers an idea of what your study is about is really important. You're also going to use it to distinguish your work from others that are similar. So if you're doing a diagnostic for Alzheimer's disease, how is that different from this other paper that was looking at a diagnostic for Alzheimer's disease? So your title needs to show how those two are different from each other. Titles can be structured in several different ways. So let's look at the types of titles. One of them is the indicative title or the declarative title. And this is a title that's in the form of a sentence, and it usually declares a result. That's why it's called a declarative title, or it indicates what a result. There's also the descriptive type of title, and this is going to describe the content of the study. And then there's a question type of title, which literally asks a question in the title. And by asking that question, it's implying a promise that they're going to answer that question in the course of the paper. There can also be compound titles. This is a title that has two parts. And any of these indicative, descriptive, or question titles can also be a compound title. Let's look at some examples of these title types. So indicative or declarative title type examples are here. A recently involved isoform of the transcription factor BES1 promotes brassinosteroid signaling and development in Arabidopsis thaliana. So you see that's a complete sentence. It has a subject, it has a verb, which is promotes, and then it has an object. So it's a complete sentence, and it's telling a result. This transcription factor promotes brassinosteroid signaling. Another example is this one. Gibberellins regulate iron deficiency response by influencing iron transport and translocation in rice seedlings. So again, that's a complete sentence, has the structure of an English sentence, and it tells a result. Some examples of descriptive titles are understanding the biochemical basis of temperature-induced lipid pathway adjustments in plants. So that's not a complete sentence, which is fine. It doesn't need to be. It's describing what's in that paper. It's describing what they're doing in this paper. They're working on understanding the biochemical basis. Another example is transposition of a rice mutator-like element in the yeast Saccharomyces cerevisiae. Again, it's not a complete sentence, but it's telling what's going to be in that paper, what the study is about. Some examples of question titles are cadmium-induced copper deficiency responses in Arabidopsis thaliana. Are phytochelatins involved? So notice that that one is also a compound title. It has two parts. So it's a compound question title. Another example is, does abiotic stress cause functional B vitamin deficiency in plants? So by asking that question, the authors are implying that they're going to answer whether or not abiotic stress causes functional B vitamin deficiency. That's kind of their research question. So how do you write a good, strong title? Well, one thing your title should be is concise, yet complete. And it's also clear. Okay, so those example titles we looked at, they were varying lengths, but none of them were really long. So they were concise, they were complete, and they were clear. So those were good titles. Now, some people are under the impression that declarative titles are the best. That's the way to go. But they're not necessarily the best titles. Don't just automatically write that type of title. A couple of things to keep in mind. Research studies that have looked at citation rate versus title types have found that colons tend to be associated with higher citations. In other words, colons indicate a compound title. So compound titles tend to be cited more, and question mark titles tend to be cited less. Why that is, no one really knows, but those are the trends from past publications. It doesn't mean that compound titles are better. It doesn't mean that question titles are worse. You can choose any of these types of titles, but it's just good to be aware of the different types and the implications that they might have on your citation rate. Additionally, shorter titles are associated with higher citation rates. So don't make your title too long. Keep it short, keep it concise. And you should avoid abbreviations in the title unless it's a standard abbreviation like DNA or PCR or something like that everyone knows or everyone should know. You can use those abbreviations, but don't use any specialized abbreviations in the title. Now let's look at some examples of titles from highly cited papers. So what I did is I went into Google Scholar and I looked at the top journals in a few different scientific fields, and then I just grabbed some titles out of those top journals. And so these are some of those titles. So in the field of ecology, here's a title, says Environmental Heterogeneity as a Universal Driver of Species Richness Across Taxa, Biomes, and Spatial Scales. So what type of title is that? That is a descriptive title. Another one is the effects of phenotypic plasticity and local adaptation on forecasts of species range shifts under climate change. Again, that is a descriptive title. Another one is how context dependent are species interactions? Question title, obviously. And then the final one here is heavy metal pollution in surface water and sediment, a preliminary assessment of an urban river in a developing country. So that is a compound descriptive title. So 3 out of the 4 here are descriptive titles. That's not necessarily representative of the entire field of ecology. These are just 4 that I grabbed pretty much arbitrarily. Let's look at some titles from biochemistry. circRNA biogenesis competes with pre-mRNA splicing. What type of title is that? That is a declarative title, right? It's a complete sentence and it's telling a result. The next one, mixed lineage kinase domain-like protein MLKL causes necrotic membrane disruption upon phosphorylation by RIP3. Again, that is a declarative title or an indicative title. Full sentence with the result. Circular RNAs in the mammalian brain are highly abundant, conserved, and dynamically expressed. Again, that's a declarative title. So in my experience in biochemistry, most titles are declarative titles when you get into the higher ranked journals. That doesn't necessarily mean they're the best. That's just what's most frequent. How about microbiology? Let's look at a few titles there. The first one we're going to look at says, revealing the bacterial butyrate synthesis pathways by analyzing metagenomic data. What kind of title is that? That is a descriptive title. Analysis of the upper respiratory tract microbiotas as the source of the lung and gastric microbiotas in healthy individuals. Another descriptive title. And finally, Middle East Respiratory Syndrome coronavirus infection in dromedary camels in Saudi Arabia. Again, another descriptive title. So I think what you can do there is just look through various titles in papers that are similar to the paper you are writing and see how they do it, see what they use, and which ones you feel like are the most effective. Now, in addition to titles, you might need to supply the journal with some keywords. Keywords are used by the journals for indexing. They might also be used by the journals to help select reviewers. But they're also used by search engines. So some of the journals, as I mentioned, are going to require you to submit a list of keywords. Some journals don't require it. If you are going to submit some keywords, make sure you select important terms. And in addition to selecting important terms, try to use keywords that are not in the title. And so that's going to provide more terms for indexing and search engines to pick up on. Also, when you're choosing your keywords, choose specific terms that are going to distinguish your work from similar papers and that are going to draw in the desired audience, just like you did when writing the title. What are some common problems of the title? Let's look at this list. One common problem is the title is too general. In other words, it's not going to distinguish what's different about your study from a similar study that was published a year or two prior. And if a title is too general, readers are not going to have an idea of what the paper is about, and they're less likely to continue reading the paper. Another common problem is that the title is too long. Now, we already talked about shorter titles are cited more frequently, so that's one reason to write a shorter title instead of a long one. Another reason is that some journals have a character limit, and you just simply cannot have a long title because the paper will not be accepted into the submission portal if the title is too long. Another common problem that I see sometimes is that the title is just too comprehensive. It gives away the entire story. You don't need to read the paper because the title includes everything. Yeah, I've seen papers where I thought, why would I even read that? Because the title tells me everything I need to know. And also, don't write a clickbait title, something that's going to overpromise what the paper delivers. You might be tempted to write a really exciting, really hyped title because you think it's going to get more readers, which may be true, but then if the readers come and they're disappointed by what's actually in the paper, if the paper doesn't live up to what the title is promising, they're not very likely to recommend that paper to their colleagues or talk about it in journal club or cite it later. So don't overpromise in your titles. Now let's get into writing the abstract. The abstract is a really important part of marketing your paper also, just like the title. Because when you go to Google Scholar and you're searching for literature to read or to cite or to learn some methods from, first you're going to see the title. Then when you click on the title, you're going to see the abstract. You're going to read the abstract. And depending on what you read there, you'll either download the paper and read it, or you'll move on to the next. So the abstract is really important in that way. It's kind of a marketing tool. It gets the readers to continue reading the paper, just like the title got the readers to continue to the abstract. The abstract will get the readers to continue to the introduction and the rest of the paper. So what are the functions of the abstract? We talked about that a little bit, but let's take a look at this list. One function is it's going to provide an overview of the paper. The second function is what we just talked about. It's going to hook the reader. It's going to convince them to go ahead and read the rest of the paper. So it should be interesting and it should be compelling in order to serve that function to attract readers. But on the other hand, it should be complete enough that somebody could cite your paper just from reading the abstract. Don't you want them to read the whole thing? Well, yeah, of course, but maybe you're publishing in a journal that's not open access and some readers cannot access the full paper. All they can access is the title and the abstract. Well, you want them to still cite your paper, so give them enough information that they can do that. Let's look at some tips for writing the abstract. The first tip is, before you start, go back and double check the instructions to authors for your target journal. That's going to tell you the format of the abstract, and it's going to tell you the word limit of the abstract. So an abstract is short, and that makes it really hard to write. Tip number 2 is that you should write the abstract just like you wrote the other sections. In other words, start from a blank screen. Number 3 goes along with that. Do not cut and paste sentences from the main text to build up the abstract. A couple reasons for that. When readers see the same sentence twice, they tend to start to tune out. They don't like to see information being repeated word for word. Secondly, it makes the abstract not flow very well if it's made up of sentences that are pulled from the body of the paper, because they're just not matching. And to make them match, you have to do a lot of editing. And so that really is probably going to take more time than if you just wrote the abstract from a blank page. Now, I mentioned before that you should look back at the instructions for your target journal to see the format and word limits of abstracts. So what are the different types of abstracts that you might write, not just when you're writing a scientific paper, but also in other aspects of scientific writing? Now, for your papers, you are probably going to write either an informative abstract or a structured abstract. The informative abstract is the type that is a single paragraph, whereas the structured abstracts contain subheadings or bullet points. And so your target journal is going to tell you which of these formats you should use. If you write a review article or a book chapter, you'll write something called an indicative abstract, which is a lot like the informative abstract, but it doesn't have all of the same elements since it's not a research study. You'll also, in your career, probably write a conference abstract or a proposal abstract, and those are going to have specific instructions for them as well. And today we're just going to focus on abstract for your journal article. For either of these types of abstracts, it's going to contain all of the elements of your journal article in that short abstract. So it's a little tricky and challenging to write. So for the informative abstracts, they're going to contain sentences that include the background, the research question or purpose, the experimental approach, the key results, not all of the results, but the key results, the main conclusions that come from those results, and also the significance of the work. So all of these elements are going to be contained in that 200 to 350 word abstract. Let's look at a template for the informative abstract based on the nature abstract. Now, these instructions are not specific only to this journal. They're going to work for any journal that uses the informative type of abstract. So you're going to start off with 1 or 2 sentences that provide a basic introduction to the field that are comprehensible to a scientist in any discipline. So very broad, context setting sentences. Then you'll have 2 to 3 sentences of more detailed background, comprehensible to scientists in related disciplines. Then you'll have 1 sentence clearly stating the general problem being addressed by this particular study, and 1 sentence summarizing the main result with the words "here we show" or their equivalent. Then you'll have 2 or 3 sentences explaining what the main result reveals in direct comparison to what was thought to be the case previously. or how the main result adds to previous knowledge. Then you'll conclude with 1 or 2 sentences to put the results into a more general context. So this is a really great template to use. I use it every time I need to write an informative abstract. Now let's look at some instructions for a structured abstract. So this particular set of instructions is from the journal New Phytologist. So these instructions are specific to this particular journal, but they give you an example of what a structured abstract is going to look like. They call the abstract the summary. So the summary for research papers, which must be usable as a standalone document, must not exceed 200 words, so that's a pretty short one, and should be organized using 4 bullet points to indicate 1. The research conducted, including the rationale, 2. Methods, 3. Key results, and 4. The main conclusion, including key points of discussion. It should not contain citations of other papers. So within these 4 bullet points, you're going to have all of the elements that you had in the paragraph type of abstract and all the elements that are in the main paper. And you're going to compress those into these 4 bullet points. So in the first bullet point, you're going to have the background, the knowledge gap, and the research question. In the second bullet point, you'll have the approach. The third bullet point will reflect the results. And then the fourth bullet point will reflect the discussion section of the paper. Let's look at one more example of a structured abstract. This is from the Journal of the American Medical Association, JAMA. So this one allows 350 words, and it asks you to use subheadings. So the New Phytologist asked for bullet points. This one's going to ask for subheadings, and those would be importance, objective, design, setting, which will all be combined under one subheading if the paper is accepted. It also has participants, interventions for clinical trials, or exposure for observational studies. Then it has the main outcomes and measures. Then it has the results, and it has the conclusions and relevance. Let's take a look at some of the common problems of the abstract. One is that there are missing elements. So make sure you have all of those required elements, the background, the knowledge gap, the research question or purpose, the approach, the results, the conclusions, and the significance. It's a lot to fit into a small space of an abstract. Or you might have those elements there, but they're not signposted clearly, and so that makes them obscured. That's a second common mistake. And a third one is over the word limits. Now, you're not going to be able to submit your paper to the journal until you have the abstract under the word limit, because with the electronic submission portals that they have nowadays, it's just not going to accept the abstract if it's over the limit. So make sure that you spend time to get it into the word limit before you go into the submission process. We're ready for the action list for this week. What I want you to do first is to brainstorm. And I want you to sit down at your computer or with a pencil and paper and just write out different versions of titles. So write 5 or 6 or even more if you want to, different possibilities for titles. So play around with the different title types. Declarative titles, descriptive titles, question titles, compound titles. Just write a whole bunch of different versions and variations, and then choose the one that you think is the best. Next on the action list is I want you to write the abstract and start with a blank page or a blank screen and write it from start to finish. You can look back at your main paper to remind yourself of how you're structuring everything, but don't cut and paste from the main paper. Write it from scratch. Depending on your target journal and which type of abstract you'll need to submit, follow some of the templates that we've looked at for informative or structured abstracts. And make sure that the abstract includes all of the key elements: the background, the research question or purpose, the approach, the key results, the conclusions, and the significance. As we wrap up this lesson, let's just look back and review the learning outcomes. When you're done with this lesson, you will be able to explain the purpose of a title. You'll be able to describe and identify the types of titles. You will be able to write a complete, concise specific title. You'll be able to explain the purpose of an abstract. You'll be able to describe all the key elements of the abstract, and you'll be able to write an abstract that contains all of the elements and conforms to the journal requirements, all while avoiding the common problems of the title and abstract. All right, that concludes this lesson on the title and abstract. Now, don't be fooled. Just because these are short parts of the paper doesn't mean that they're easy parts of the paper. Put a lot of thought and effort into writing the title. Put a lot of thought and effort into writing the abstract. Your first attempt at the abstract is probably going to be too long, and that's okay. You'll revise it, you'll edit it until you get it into the word length. So go ahead and get started on this week's action list, and happy writing. Welcome to the lesson on revising your manuscript. We're going to talk about getting to the final version, and then we're going to talk about getting ready for submitting your paper. Let's start off by taking a look at where we are on our project management diagram. So we are finally getting to the closing part of the project. We've gone through initiating, planning, and executing, and now we're ready for closing. Let's take a look at the learning outcomes for this lesson. At the conclusion of this lesson, you will be able to revise your manuscript for structure, cohesion, and flow. That's looking at the overall manuscript. You will also revise each section of your manuscript, edit your writing for grammar, spelling, and style. You'll know what to do to write a cover letter for submission to the journal of choice, and you know how to prepare for the submission process. First, let me say congratulations that you've gotten to this stage. So if you're seeing this lesson and you've completed all the previous lessons, you have a full first draft. You know, that's a big accomplishment, so congratulations. If you have time at this stage, what I would like you to do is just go away for a few weeks. Just let your manuscript sit. Sit in your computer, back it up, and make sure you have a backup copy of it. But just don't read it, don't work on it, don't edit it. Don't even think about it if possible. Think about other aspects of what you need to do and just let it sit. Then when you come back to it, you'll have what we call fresh eyes. So you'll be able to read the paper with fresh eyes, and you're going to notice things that you didn't notice before. Now, sometimes you don't have time to step away. You need to keep working quickly, and that's okay too. I think it's better if you can take a break and then come back to it. But if you don't have time, you just don't have time. So you can keep working steadily. Now, when I say revising and editing, what do I mean? Let's take a look at some definitions of these two terms. Well, revising is when you are taking another look at your ideas, you're adding information, you're cutting information, you're moving sentences or paragraphs around, and you're making changes to make the writing more clear, accurate, interesting, or convincing. So what is editing? Editing is when you're adding or changing words. You're fixing problems with grammar, punctuation, sentence structure, and style. So revising is more high-level modifications to your paper, and editing is more at the technical and specific level on the paper. And in practice, you might be doing them simultaneously. I recommend in the course that you do the revising part first and then come back and do editing. And that's because it's going to be more efficient to revise first and then edit later. If you're editing at the same time that you're revising, you might lose track of the big picture. The overview, the looking out the airplane kind of view that you need to have when you're revising, because editing is pulling you down into the weeds, into the very specific details. Now, instead of concluding this week's lesson with an action list, the lesson is going to be an action list. There's not a lot of conceptual things for me to talk about today. It's mostly action. So we're going to go step by step, and you have a download in your course materials that is a master checklist. We're going to look at a series of individual checklists, and that download is going to have all of those checklists on one piece of paper, and you can print that out and mark it off as you go. Okay, so let's get started. Action item number 1 is to start revising. And you're going to do this at the high level overview first. So what I want you to do is sit down and read the entire draft. And as you're reading it, think about the content and the location of that content. In other words, notice all of the key elements, all of your logical arguments and conclusions, and where you have put those. Also, what I want you to do while you're doing this reading and noticing the content is to make a reverse outline. So get a piece of paper and a pen or a pencil and just start labeling it paragraph 1, paragraph 2, paragraph 3. And as you read that paragraph, write down what is the key message in that paragraph. And as you do this for the entire paper, it's going to help you see how you have structured your paper. You might feel like you already know how you structured the paper, but sometimes taking a look afterwards, in reverse, doing a reverse outline, it's going to look different than your forward outline. And this will help you notice when you need to move big chunks of text or information to a different place within the manuscript. Okay, so let's start with the first checklist. This is the revisions checklist for your first pass overview revisions. And these prompts that you'll see are usually set up as a yes or no question. But I don't want you to just answer it yes or no. These are not literally yes or no questions. They're more prompts to get you to think about how you have written the paper and think about what changes you might need to make improvements on how you wrote the paper. So the first thing on the checklist is, are the paragraph contents consistent? Is there consistency between paragraphs and sections? So again, don't just say, oh yes, they are consistent. No, you need to think about it as you read it and think about whether there is that consistency within and between the paragraphs. The second point is, have you made the reader's job easy, or do they have to look up additional sources for information? So, that might apply especially to the materials and methods section, but just think about that in the entire paper. Is everything that you have stated correct? Is it factually correct? And you might need to verify. Sometimes if you're trying to write quickly, you'll just write something down from memory, and maybe you got a detail a little bit wrong. So this is a good time to do your fact checking. Verify that each reference you're citing actually says what you say it's saying. If your writing has factual errors in it, you're going to lose a ton of credibility with your readers. If you get one fact wrong in the introduction, they're going to question everything else you say for the rest of the paper. So make sure everything is correct. Next, ask yourself, is the manuscript cohesive between paragraphs and between sections? So this is a little bit different than consistent. Cohesive and consistent are not quite the same. Cohesive means, is there flow? Does one paragraph logically lead into the next paragraph? Or does it feel like it's jumping around all over the place? Ask yourself, does the writing clearly convey the ideas that are being presented? So is the writing clear? Is it understandable? Or do you need to revise some sentences so that clarity is improved? Ask yourself if the manuscript is complete. Did you miss something? Do you need to add some additional information to make it complete? How about the length? Can the manuscript be made more concise? Is it too wordy? Is it too long? How can you tighten it up and make it concise? Going back to the concept of consistency, what about your key terms? Are you using them the same way every time throughout the paper? When you have lists, are they structured in a parallel format? Are the words and phrases that you're choosing, are they precise? Or is there some room for interpretation there? Is there some ambiguity or some lack of detail? And also ask yourself, is this convincing? Are my arguments convincing? Will the readers believe what I'm saying and want to act on it? And if you feel like it's not convincing, well, how can you make it more convincing? Maybe that is looking up additional references that can be cited to make a stronger case for what you're trying to say. Maybe that means discussing some of the figures a bit more thoroughly So whatever you need to do to make it more convincing, go ahead and do that during this revision stage. Okay, so that's the overview revisions. Now we're going to start looking at each section and revising each section more specifically. So this will be your second pass of revisions. And the introduction might need a lot of attention. That's normal at this stage, that the introduction is going to need more work than some of the other sections. Because now that you have written the discussion where you synthesized all of your results and you put it together with comparison and contrast to other previous studies. And so maybe when you go back and read the introduction, it's not really setting up for the discussion all that well. So if your introduction is no longer a really good setup for the discussion section, this is a good time to notice that, and you can make those changes in the introduction. So let's go through the checklists for each of the sections. Now we're going to start with the title. Ask yourself, does the title reflect the research question or purpose and its answer? Is the title concise, with no unnecessary words or phrases? Will the title compel readers to read the abstract? Does the title state the main topic of the study? Will selected keywords increase search engine hits beyond words in the title? Is the title clear and unambiguous? Have abbreviations been omitted except for common terms? Is the title complete? Does the title separate the article from other articles in the field? And you'll also need to submit with your journal article manuscript what they call a running title. And this is the short title that you'll see at the top of the second page, third page, and so on. And does that running title reflect the full title? Is it recognizable as a shorter version of that full title? Let's look at some prompts that are the checklist for revising the abstract. You want to ask yourself, is the research question or purpose stated? Is the approach stated? Are the key results stated? Is the research question or purpose answered? Are all of the elements signposted? Is the abstract within the word limit? Is the significance clear? Is the context clear? And does the abstract adequately summarize the paper? And one way to answer that would be to ask yourself, could a reader cite my paper just from reading the abstract? All right, how about the checklist for the introduction? And as I mentioned, the introduction might be a section that you have to revise more heavily than you have to revise the other sections. So here's your list of prompts. First, does the background funnel to the research question? Is it not a literature review? Remember, it should not be a literature review, it should be a background. Secondly, are references used when needed, and are they placed correctly within the sentence? So you don't automatically put the reference at the end of the sentence. Sometimes it's in the middle of the sentence. Also ask yourself, is the research question stated clearly and precisely? Do all of the components logically follow each other? Have all elements been clearly signposted? So again, it's important to let the readers know when you're giving them a key piece of information with signposting words or signaling words. Next, ask, is the approach mentioned in the final paragraph? Now again, this is optional, but I think it's a good idea to mention the approach in the final paragraph. Ask yourself, are the key results or conclusion mentioned in the final paragraph? Is the significance or implication stated clearly and precisely? Looking at the overall introduction, is it cohesive and coherent? In other words, are the paragraphs flowing from one to the next, or do they feel like they're jumping around all over the place? And does it make sense? Is it coherent? Is it too long or is it too short? Make sure it is a good length for papers in your field, in your discipline. Let's look at the checklist for materials and methods. So, are all the materials described correctly and with enough detail? So if you need to have the manufacturer, the chemical name, whatever biologicals you used, so on and so forth, do you have all of that detail included? Are all of the methods described correctly and with enough detail? So make sure that you look for typos in your methods. If you remember my story about the decimal place being off in a nutrient solution that caused a lot of problems for my student. You can see the importance of not having mistakes in the methods. And also make sure you have enough detail. So do you talk about the important conditions for doing this procedure, like the temperature, the pH, the volume, the concentrations of various chemicals? Have you used references when they're needed? And are the references placed correctly within the sentences? Have you organized the materials and methods section logically? So using logical subheadings and putting them in an order that makes sense to the readers. If you have any new or unusual procedures, have you stated the purpose for those? Is the section free of material from other sections? In other words, have you excluded any introduction or discussion or results from this materials and methods section? As we mentioned before, passive voice is preferred in the materials and methods section. So is the voice primarily passive voice? And did you use verb tenses correctly? It should be mostly past tense because you're describing things that you did in the past, but there might be sentences where you describe the way things are as an explanation for why you did certain procedures, and that might be in the present tense. And then lastly, ask yourself, are the word choices precise? Now let's look at the checklist for the results section. So what you want to do is ask yourself, is the results section organized in a specific way that makes a lot of sense? Such as from the most important results to the least important results. That's one way that you could go about it. And also within the paragraphs, how have you organized the paragraphs? Are they organized from the most important to the least important? When you have statistical data such as a p-value, is that information written such that it's supporting the data, but that p-value is not presented as the result itself? So it's supporting the result, it's not the result. Ask, have you directed the reader to figures and tables within a result rather than just announcing, go look at this figure or go look at this table, or announcing what's in the figure or table? So you want to do that skillfully and present the direction to the figure within a result. Ask yourself, are the results explained in enough detail? And have I included the control results, the positive controls and negative controls if you have those two types of controls? Is the section free of material from other sections? So does it have discussion material in it? That's the most common problem. You might have materials and methods material in the results section. So make sure that results is purely results. Check whether you have used the repeating pattern. If you're going to use this pattern, that is having the purpose, approach, result, and interpretation for each of the results. Are irrelevant statements and tangential information avoided? So are you keeping focused? Are you focusing entirely on describing the results? Are the references placed correctly and where needed? There's not going to be very many references in the results section, but you might have 3 or 4 scattered about. So make sure if you do have references, they are used properly. Are the most important results emphasized? So can the reader distinguish the most important results from minor results? And think about the tenses. Have you used tenses correctly? Again, in the results section, most of the tenses should be past tense. Only rarely will you use present tense in the results section. Now we get to the discussion checklist. So what are the questions you need to ask yourself about writing and revising the discussion section? Well, first ask, is the section well organized? Such as, from most important results to least important results. Are the results compared and contrasted with results of previously published studies? Now that is opposed to just mentioning the previous published studies and citing it without doing the work of comparing and contrasting and explaining how your research relates to those previous results. Think about, are discrepancies, unexpected findings, and limitations discussed? Is a conclusions paragraph at the end of the discussion, and is that paragraph not a summary? So again, conclusions and summary are not the same thing. You want to do conclusions, you don't want to do summary. Does the discussion follow a pyramid structure? Is the answer to the research question in the first paragraph? That's where it should be. Is the answer followed by supporting evidence? And that supporting evidence will be partially your own results, and partially it might come from some of the previous studies. Is the section free of material from other sections? In other words, have you excluded introduction material from the discussion? Have you excluded results material from the discussion? Is the significance of the work clearly discussed? And are the findings generalized to other use cases? So it is important to generalize your results in the discussion section. In other words, showing how the results that you found in your specific study can apply to other situations, other organisms, other locations, other diseases, whatever the case might be. And have you avoided repeating the results material? And have you signposted all of the elements? All right, so after you've done this, after you've gone through all of your sections and made revisions, now you can come back and start to edit. So you can start to edit for grammar, edit for spelling mistakes, if there are any still remaining, and edit for style. So here's your editing checklist. First, ask yourself, is the grammar and technical style, such as person, voice, tense, pronouns, prepositions, articles, is this all correct? Is the word location correct, such as, is the verb following the subject? Or did you put the old and short information at the beginning of a sentence? Have you removed unnecessary items, redundancies, and jargon, for example? Are the non-technical words and phrases that you are using, are those simple? They should be. So don't overcomplicate things. Don't try to use fancy language when it's not necessary. Just go ahead and use simple language, plain language. That's fine. In fact, that's better than using fancy and flowery language. Have you found all of those spelling and punctuation errors? Are comparisons written correctly? And so in this case, we're not talking about compare and contrast to previous work. We're talking about comparisons, well, it could be, but we're mainly talking about comparisons within a sentence, such as our results are similar to results from Smith et al. 2012. That is a correct comparison, but an incorrect comparison would be something like, our results are similar to Smith et al. 2012. So you're comparing results to people. That's not a good comparison. So you need to compare results to results. Have you eliminated and resolved any noun clusters? So noun clusters are a series of nouns that are strung together, and it's really hard to read those. Are transitions used, and do they make sense? Is the action in the verbs? Have you avoided nominalizations? So if you don't know what a nominalization is, go ahead and look that up. And if you find them in your work at this stage, this is the time to change the sentences around so that you're using verbs as verbs instead of nouns as verbs. Also ask yourself, is sentence length varied, and do you have one idea per sentence? And then also at this stage, you need to verify the format and the completeness of the references that you have cited. So way back in the lesson when we talked about finding and citing references, I mentioned that in your reference managing software, you might have to make some corrections because certain sources of citations are not formatted correctly. For example, Google Scholar, I have noticed, is often very bad about capitalizing all the words of the name of a journal. You might have a journal such as the Journal of Experimental Biology, and only the word journal is capitalized. They don't have experimental or biology capitalized, but they should be. So you'll have to go in and make that correction. So look at all of your references, make sure they're all correct. If they're not, correct that in your database. Now, some of these things that I've talked about here, if you want to learn more about those, then you can take my other course on scientific writing skills. So it's a complement to this course. It's not required to do this editing, but if you want to do a really great job of editing, that's a course that's going to help you make your writing the best it can possibly be. All right, so now you have revised your manuscript, you have edited your manuscript. this is the time to send it to your co-authors. And so your co-authors will give you their suggestions. They'll probably do some revisions themselves, and they'll probably do some edits themselves. And then they'll send it back to you. And you may have to do this process 3 or 4 times, or maybe even more. So just be prepared for that. It's very rare to just do one round of revisions and edits and be done. You'll typically do several rounds. This is also the time that if you can, if your work is not something that's too proprietary or you need to keep it completely secret, this is a good time to have your manuscript reviewed by 1 or 2 people who are not involved in the project. So some friends of yours who are willing to do you a favor, or some colleague. And if they're friends of yours and they're in a slightly different field and they don't understand the science, that's okay. Actually, that might even be better, because then they'll be reading it for a different purpose than somebody who's in your field. So that person who's in a different field is going to notice different things that the person who's in your same field and is going to read it for the scientific aspects, they might miss some of the writing aspects. And when you give your paper to these outside reviewers, ask them to be critical of it. In a constructive way, of course. So constructive criticism is going to help you, because it might prepare you to make some changes that reviewers would have picked up on if you had submitted it without this constructive criticism. Now, one important thing I want to mention is take all of the comments very seriously. I've seen a lot of writers, they'll get a comment from somebody and they'll just say, oh, well, that person just didn't understand what I was saying. And that's exactly right. They didn't understand what you were saying, but don't blame them. If they don't understand what you're saying, it's because you didn't write it as well as you could have. So take that criticism very seriously and think, try to step back into the reader's position and see it from their point of view. And then that will help you revise and edit so that those kinds of questions don't come up. Okay, so you've had your paper reviewed by your co-authors and you've had it reviewed by some outside reviewers. Now it's time to prepare for submitting it to your target journal. All right, we're almost there. So the first thing you want to do is make sure that all of the authors have approved the final version. Get their specific approval. Yes, it is ready to submit. You want that in writing, if at all possible, by email, for example. Make sure the manuscript is formatted to the journal's specifications. So they're going to tell you how to structure the title page. They're going to tell you where to put the figure legends, if that's going to be with the figures or if it's going to be in the manuscript after the references. They'll tell you all of those different formatting things that you have to do. So make sure you have those instructions and you followed them. Number 3, write a compelling cover letter to the associate editor. And we'll talk about more details about the cover letter soon. Number 4, you will upload all of the files to the journal's submission portal. It might take several hours to do this upload. So be prepared for that. Set yourself up with a big block of time so that you don't get interrupted. I used to do it in the evenings or on the weekend because there were fewer people around that would come into my office and ask me questions during that time. And number 5, only submit to your target journal. Don't submit to multiple journals at the same time. And by the same time, I don't just mean the same day. I mean, if you're waiting for 5 or 6 weeks for a decision from one journal, you cannot submit your paper to another journal during that time. You have to wait until you have a final decision. And if that final decision is a rejection, then at that point you can submit to a second journal, but not at the same time. If you do submit to multiple journals at the same time, they're going to find out, and then both or all of those journals is going to kick your paper out and you won't get it reviewed. So what about this cover letter to the associate editor? What is the purpose of that? So the purpose of the cover letter is kind of like the purpose of a title or an abstract. It's to get the associate editor interested in your paper and to show them that your paper is a good fit for their journal and that the readership of that journal will be interested in your paper. If the associate editor feels like all of that is true, then they'll send the paper into the peer review process. So how do you write this cover letter? Let's look at an example of instructions from the journal New Phytologist. So they say that authors should include answers to the following questions with a maximum of 50 words per question in a covering letter to help editors decide whether to send the manuscript for peer review. And those questions are: what hypotheses or questions does this work address? How does this work advance our current understanding of plant science? Why is this work important and timely? So I think this is a really great template for a cover letter to any journal. So if you're not doing plant science, let's say you are in microbiology, just switch out the words in the second bullet point from plant science to microbiology. This slide is here as a reminder for the decisions that might come out of the review process. And just be prepared for that. You're probably going to have to address reviewer comments. And we already talked about in an earlier lesson addressing the reviewer comments and how to do that. Congratulations! Once you have done all of these steps, you are ready to submit your manuscript. After you submit, I think this is a great time to celebrate and reward yourself because you've reached an important milestone. Even though you haven't got the decision yet, still, this is a really big milestone. It's a big step in the process. You've written your entire paper, you've revised it, you've edited it, you've submitted it. Yeah, it's time to take a little, victory lap. Get an ice cream cone or a pizza or, however you like to reward yourself. Have a movie night, go to the park, whatever you think is a good reward. So that concludes the lesson on revising, editing, and submitting. This is basically the end of the course. All right, so as you prepare to submit your journal article, I would say once again, happy writing.